Am I audible and visible? Let me check. Yeah. If I'm audible and visible, can I see a thumbs up, please? Hi, Himanshu. Hi, Shivangi. So welcome to this FMGE 2026 High Yield Biochemistry Last Resort Revision. In this session, yes, this will be a bit lengthier session. Probably we will be discussing for the next 4 hours. And in the next 4 hours, I will be covering all topics in biochemistry. I've chosen high yield previous year MCQs, and all these MCQs will be discussed conceptually. And today's session, I have focused on genetics because in the last session, which we conducted live, many of you had asked me to discuss about genetics. So in this session, we'll be discussing about genetics and applied aspects of genetics. Yeah. Yes, INICT, NADPH, all students can watch this because there is no difference between the topics that need to be covered for FMG and the topics that need to be covered for NADPH or INICT. The topics remain the same. The way I discuss would also be the same. Probably the type of question that is asked can be different, right? Is there a disturbance? 1 second. Let me switch off the fan. Is it clear now? I hope the voice is clear now. Okay, good. It's fine. So let's start this. And this session, there are 6 sections. The section number 1. 1 second. So section number 1 is about cells, suborganelles and tests. Okay. So this is the first question. This FMG Jan 2023 MCQ. Which of the following pathways only takes place in a cell cytoplasm? Can you all start answering? I would want this session to be an interactive session. What do you think is the right answer? The right answer is glycolysis, right? So I want you to know that glycolysis is the only pathway that can generate ATP even in the absence of oxygen. Any pathway which is not dependent on oxygen happens in cytoplasm. If it is dependent on oxygen, it happens in mitochondria. Do you all understand this? So glycolysis happens in cytoplasm, whereas beta oxidation happens in mitochondria. Just because I said beta oxidation happens in mitochondria, it does not mean all fatty acids get oxidized in mitochondria. There is an exception. Very long chain fatty acids get oxidized in peroxisomes. All of you please remember all these exceptions. Very long chain fatty acids get oxidized in peroxisomes. That means you will have to know all functions of peroxisomes because there was a previous year question asked in FMGE about functions of peroxisomes. So first function is there in the slide. It is related to very long chain fatty acid oxidation. It is also related to branch chain fatty acid oxidation. It is related to ether lipid synthesis. Okay, it is also related to ether lipid synthesis. This is where there is glycine and taurine conjugation of bile acids. So what are the functions of peroxisomes? Peroxisomes are related to very long chain fatty acid oxidation, branch chain fatty acid oxidation, ether lipid synthesis, glycine and taurine conjugation of bile acids. Okay, so that's about this choice B, beta oxidation. Where does citric acid cycle happen? Obviously, citric acid cycle happens in mitochondria. How about urea cycle? Urea cycle is a pathway which happens partly in mitochondria and partly in cytoplasm. So all of you please remember there are 2 pathways which happen both in mitochondria and in cytoplasm. One is urea cycle, the other one is heme synthesis. Okay, good. So can we fill up this table, all of you? Please start answering. Where does glycolysis happen? So this is going to be a high yield conceptual session. And first of all, my sincere apologies for the delay in the start of the session. It was a technical issue, right? For some reason, my iPad was not shown on OBS. So it took some time. Otherwise, this is going to be a high yield session. Every minute of your presence here counts. I'm not going to talk about anything else other than biochemistry, and it's all about high yield facts. So I want you to stick to this session. Okay, so do not leave the session. Be here for the next 4 hours. You will then come back and thank me. Okay, so glycolysis. What is glycolysis? Glycolysis is glyco plus lysis. It is a process by which you split a 6-carbon compound called as glucose to form 2 products. And the 2 products differ depending upon whether the process occurs aerobically or anaerobically. If the process occurs aerobically, products formed are 2 molecules of pyruvate and 7 molecules of ATP. This can be a one-liner. All of you tell me, what are the products of aerobic glycolysis? 2 molecules of pyruvate and 7 molecules of ATP. When the same process happens in the absence of oxygen, products formed are 2 molecules of lactate and 2 molecules of ATP. So what are the products of anaerobic glycolysis? 2 lactate and 2 ATP. That is why I told you glycolysis is the only pathway that can generate ATP even in the absence of oxygen. Because it is not dependent on oxygen, it need not happen in mitochondria. It happens in cytoplasm. Got it? So glycolysis happens in cytoplasm. Now, what is PDH complex? If you watch this session for the next 2 to 3 minutes, once we fill up this entire table, you will have an overall understanding of biochemistry. So watch this carefully. PDH complex. PDH complex is a link between glycolysis and citric acid cycle. Do you understand this? PDH complex is a link between glycolysis and citric acid cycle, because what did I tell you happens in glycolysis? In glycolysis, 1 glucose becomes 2 pyruvate. After this, these 2 pyruvate should enter into citric acid cycle, because only when something enters into citric acid cycle, it comes out as carbon dioxide, which can be exhaled out. So how does pyruvate enter into citric acid cycle? By becoming acetyl-CoA. All of you remember, pyruvate enters into citric acid cycle by becoming acetyl-CoA. Which enzyme converts pyruvate into acetyl-CoA? That is pyruvate dehydrogenase complex. So those of you who are watching this session, do not forget this. Pyruvate dehydrogenase complex converts pyruvate into acetyl-CoA so that this acetyl-CoA can enter into citric acid cycle. We all know citric acid cycle happens in mitochondria. So where is pyruvate dehydrogenase complex also present? That is also present in mitochondria. Are you clear about this? So glycolysis happens in cytoplasm. PDH complex is present in mitochondria. Citric acid cycle happens in mitochondria. Got it? Very good. So tell me, glycolysis happens in cytoplasm, PDH complex in mitochondria, citric acid cycle in mitochondria. Glycogen metabolism. Whenever I talk about glycogen metabolism, I include glycogen synthesis and glycogenolysis. Both glycogen synthesis and glycogenolysis happen in cytoplasm. Please don't forget this. Both glycogen synthesis and glycogenolysis happen in cytoplasm. Gluconeogenesis. It is quite complex, because gluconeogenesis is a reversal of glycolysis. Do you understand this? What is gluconeogenesis? It's a reversal of glycolysis. What is glycolysis? I told you, in glycolysis, you start from 1 molecule of glucose. You split this 1 molecule of glucose into 2 molecules of pyruvate. So what is gluconeogenesis? You start from 2 molecules of pyruvate and you end up in 1 molecule of glucose. Okay, so this is essential because I saw 2 questions based on this in the previous MCQ, previous paper. Okay, so what do we do in gluconeogenesis? You start with 2 molecules of pyruvate and you synthesize 1 molecule of glucose. That is done by pyruvate carboxylase. So tell me, which is the first enzyme of gluconeogenesis? The first enzyme of gluconeogenesis is pyruvate carboxylase, which converts pyruvate into oxaloacetate. So PDH complex converts pyruvate into what? All of you type it in the chat box. PDH complex converts pyruvate into acetyl-CoA. Pyruvate carboxylase converts pyruvate into oxaloacetate. Both are present in mitochondria. So the first step of gluconeogenesis happens in mitochondria. After which reversal of glycolysis happens in cytoplasm until you form glucose 6-phosphate. Now tell me, what will be the enzyme which converts glucose 6-phosphate into glucose? The enzyme which converts glucose 6-phosphate into glucose is obviously glucose 6-phosphatase. And that is present in endoplasmic reticulum. Glucose 6-phosphatase is present in endoplasmic reticulum. So when I ask you, in the sense when the question paper asks you, where does gluconeogenesis happen? What should be your answer? Gluconeogenesis happens first in mitochondria, then it happens in cytoplasm. Finally, glucose 6-phosphatase is present in endoplasmic reticulum. Why am I stressing so much on glucose 6-phosphatase? Defect of glucose 6-phosphatase causes von Gierke's disease. We will be learning about glycogen storage disorder in the next 1 hour. I will ask you what is the significance of glucose 6-phosphatase. You should tell me it converts glucose 6-phosphate into glucose and it's an enzyme involved in gluconeogenesis. It is present in endoplasmic reticulum. Are you clear? Very good. Excellent. So gluconeogenesis involves multiple suborganelles, mitochondria, cytoplasm and endoplasmic reticulum. Okay. HMP shunt happens in cytoplasm. Okay. HMP shunt happens in cytoplasm. Fatty acid synthesis also happens in cytoplasm. But what about fatty acid oxidation? Didn't I tell you? Fatty acid oxidation happens in mitochondria. Very long chain fatty acids get oxidized in peroxisomes. Right? Very long chain fatty acids get oxidized in peroxisomes. Cholesterol synthesis. Just remember, cholesterol is a steroid. Right? Cholesterol is a steroid. Steroids get synthesized in smooth endoplasmic reticulum. All of you please remember, rough endoplasmic reticulum is for protein synthesis. Smooth endoplasmic reticulum is for steroid synthesis. S for S. Smooth endoplasmic reticulum is for steroid synthesis. So cholesterol synthesis initially happens in cytoplasm, then it happens in smooth endoplasmic reticulum. Okay. Ketone body synthesis. Just remember, ketone bodies are products of fatty acid oxidation. Do you all understand this? What are ketone bodies? They are products of fatty acid oxidation. We know fatty acid oxidation happens in mitochondria. So ketone bodies get synthesized in mitochondria. Okay. Bile acid synthesis. What are bile acids? Bile acids are derivatives of cholesterol. Please remember, what are bile acids? Derivatives of cholesterol. Bile acid is also a steroid. So all of you tell me, where are bile acids synthesized? When I say it's a steroid, it should be synthesized in, please keep the live chat active. It gets synthesized in smooth endoplasmic reticulum. Okay. Urea cycle. Didn't I tell you urea cycle happens partly in mitochondria, partly in cytoplasm? And which is the other pathway which I told you happens partly in mitochondria, partly in cytoplasm? Heme synthesis. So this can be a simple one-liner. What are the pathways which happen both in mitochondria and in cytoplasm? Urea cycle and heme synthesis. So for the benefit of all of you, what are we going to do now? We are going to repeat all that we discussed just now. Can we do that? Yeah, all of you please quickly answer because it's a rapid revision session, right? It's a last resort revision. Revision is all about repetition. So all of you repeat it along with me. Where does glycolysis happen? Glycolysis happens in cytoplasm. PDH complex is present in mitochondria. Citric acid cycle, all of you answer. Citric acid cycle in mitochondria. Glycogen metabolism, both glycogen synthesis and glycogenolysis happen in cytoplasm. Gluconeogenesis, please answer. Gluconeogenesis, reversal of glycolysis. Pyruvate carboxylase is present in mitochondria. The remaining steps happen in cytoplasm. Glucose 6-phosphatase is present in endoplasmic reticulum. Okay. HMP shunt happens in cytoplasm. Very good. I am happy that the chat box is active. Good. Fatty acids get synthesized in cytoplasm. Fatty acids get oxidized in mitochondria. Very long chain fatty acids get oxidized in peroxisomes. Cholesterol synthesis. Cholesterol is a steroid, gets synthesized in smooth endoplasmic reticulum. Initial few steps in cytoplasm. Ketone bodies are products of fatty acid oxidation. So ketone body synthesis also happens in mitochondria. Bile acids are derivatives of cholesterol. It is also a steroid, gets synthesized in smooth endoplasmic reticulum. Urea cycle happens partly in mitochondria and partly in cytoplasm. And what is the other pathway which happens both in mitochondria and cytoplasm? That is heme synthesis. Okay. So we know the answer for this question is glycolysis. Answer this please. Mitochondrial matrix enzymes. Yeah. So did I tell you citric acid cycle happens in mitochondria? Okay. And one of the enzymes of citric acid cycle is citrate synthase. So which is the mitochondrial matrix enzyme? It is citrate synthase. Okay. Monoamino oxidase. We can write monoamino oxidase as MAO. That you can use to remember that it is present in outer mitochondrial membrane. I hope you all remember your mitochondria has got an outer membrane, it has got an inner membrane, and then there is a matrix. For the outer membrane, remember it is OM and the enzyme is monoamino oxidase. For the inner membrane, it is an electron transport chain complex. Right? What is the marker enzyme for inner membrane? It's one of the complexes of electron transport chain. It is complex 2 or succinate dehydrogenase, or it is complex 5 which is ATP synthase. So tell me which is the marker enzyme for inner mitochondrial membrane. It is complex 2 or succinate dehydrogenase, or it is complex 5 or ATP synthase. Okay. So now you know ATP synthase is the enzyme for inner mitochondrial membrane. Catalase is present in peroxisome. Okay, catalase is a marker enzyme for peroxisome. Peroxisome is so called because it keeps generating hydrogen peroxide. To detoxify hydrogen peroxide, peroxisome has catalase enzyme. So catalase is the marker enzyme for peroxisome. Is that clear? Okay. So can we learn about all marker enzymes of all the suborganelles? Yeah. So whenever you learn about one question being asked about marker enzyme, you should learn about marker enzyme of all suborganelles. Okay. So all of you start answering. Which is the marker enzyme for nucleus? All of you, do you know that within nucleus we have chromosomes? Yeah, within nucleus we have chromosomes. Chromosomes are nothing but double-stranded DNAs. A double-stranded DNA can perform replication and it can perform transcription. Replication is done by DNA polymerase. Transcription is done by RNA polymerase. Okay, we will be learning about replication and transcription in another 2 hours. right? So we are going to learn in detail about genetics today. So all of you stay with me. I'm going to tell you in detail in the sense starting from the very basic fact, all based on MCQs, okay? Nothing redundant. I have chosen all previous year MCQs. Using those MCQs, we are going to discuss all about genetics today. Okay, for now, just remember nucleus is where you have your chromosome. One chromosome is one long double-stranded DNA. DNA can perform replication and transcription. Replication is done by DNA polymerase. Transcription is done by RNA polymerase. So the marker enzyme for nucleus is DNA or RNA polymerase. Okay, endoplasmic reticulum. Can you all answer what is the marker enzyme for endoplasmic reticulum? Didn't I tell you glucose 6 phosphatase is present in endoplasmic reticulum? So the marker enzyme for endoplasmic reticulum, very good. Should we check? Yes. No, I have covered almost all topics here. Yeah, please watch this. So glucose 6 phosphatase, all of you. Glucose 6 phosphate is a carbohydrate. Glucose 6 phosphatase is an enzyme. So marker enzyme for endoplasmic reticulum is glucose 6 phosphatase. Okay. Golgi complex, G for G. For Golgi complex, the marker enzyme is glucosyl transferase or galactosyl transferase. So tell me which is the marker enzyme for Golgi complex. It is glucosyl transferase or galactosyl transferase. Now you have to answer. I've already given you the answers. For outer membrane, what are the marker enzymes? Monoamino oxidase. For inner membrane, electron transport chain complex. So what are the electron transport chain complexes that I told you? It is complex 2 or succinate dehydrogenase, or it is complex 5, which is ATP synthase complex. That is about marker enzyme for inner membrane. Lysosomes. Lysosomes are loaded with cathepsins. Okay, lysosomes are loaded with cathepsins. Cathepsins are proteases, and those are the marker enzymes for lysosomes. Yeah. Now cytoplasm. Did I tell you cytoplasm is wherein glycolysis happens? Right? Cytoplasm is wherein glycolysis happens. And I said in glycolysis, glucose becomes 2-pyruvate. And if there is no oxygen, I said 2-pyruvate become 2-lactate. The enzyme which converts pyruvate to lactate is lactate dehydrogenase. And that lactate dehydrogenase is present in cytoplasm. So all of you, I'm going to ask you 3 questions. I want the chat box to be active. Everybody please start answering. Pyruvate is converted to acetyl-CoA by which enzyme? Pyruvate dehydrogenase complex. Pyruvate is converted to lactate by lactate dehydrogenase. Pyruvate is converted to oxaloacetate by pyruvate carboxylase. Are you clear? No confusion here. So lactate dehydrogenase is the marker enzyme for cytoplasm. Now the last one is peroxisome. What did I tell you about peroxisome? Peroxisome is so called because it keeps generating hydrogen peroxide. To detoxify hydrogen peroxide, it has got catalase enzyme. So that is the marker enzyme for peroxisome. Got it, everybody? Good. Excellent. So that's about marker enzymes for all these suborganelles. Okay. So what is the right answer here? The right answer is the marker enzyme for cytoplasm is lactate dehydrogenase. The marker enzyme for peroxisome is catalase. For mitochondrial matrix is what they asked you. For mitochondrial matrix, it is citrate synthase. Okay. Now, July 2024 MCQ. Zellweger syndrome is due to which of the following? Right? It's a peroxisomal biogenesis disorder. All of you please remember what is Zellweger syndrome. Peroxisome is not even generated. The cell does not have peroxisome. When the cell does not have peroxisome, what are the functions which cannot happen? Very long chain fatty acids do not get oxidized. Branch chain fatty acids do not get oxidized. ether lipids cannot be synthesized. Okay, and this Zellweger syndrome is otherwise called as cerebrohepatorenal syndrome. It is otherwise called as cerebrohepatorenal syndrome. So all of you please memorize it. What is Zellweger syndrome? It is peroxisomal biogenesis disorder, and it is because of a defect of POX gene. In this case, what will not happen? As I told you, very long chain fatty acids, branch chain fatty acids do not get oxidized. There is no ether lipid synthesis. It is otherwise called as cerebrohepatorenal syndrome. Okay, good. Now, answer this question. This was July 2024 MCQ in FMGE. Did you all see this MCQ? Which of the following substances is used for sample collection in tube for blood alcohol concentration measurement? The answer is sodium fluoride. Choice B is the right answer. Sodium fluoride plus potassium dichromate. Okay, you need to know why. Whenever blood alcohol has to be estimated, the sample after collection should not generate alcohol. How will a sample start generating alcohol? Whenever glucose is metabolized, it can generate alcohol. So you should avoid glucose metabolism. How can you avoid glucose metabolism? By adding sodium fluoride. So sodium fluoride, do you all know that it inhibits enolase? Even if you don't remember, please memorize it now. Sodium fluoride inhibits enolase. Glycolysis does not happen. So new alcohol will not get generated. Another source of alcohol in the blood sample after collection is if microorganisms grow in the blood sample, these microorganisms can also generate alcohol. So you need an antibiotic. You need something which prevents microbial growth. That is why you add potassium dichromate. Do you all understand this? So what is the anticoagulant of choice for blood alcohol estimation? It's a combination of what? Sodium fluoride and potassium dichromate. Usually what we do is we collect blood sample in a gray top tube. What does a gray top tube have? I told you G for G. Gray top tube is for glucose estimation. It has sodium fluoride and potassium oxalate. Right, it has got sodium fluoride and potassium oxalate. That is what we generally do. Gray top tube used for glucose estimation has sodium fluoride and potassium oxalate. We collect blood sample for alcohol estimation also in a gray top tube. But if it's a severe or if it's a high level or high stake forensic case, okay, high stake forensic case when you do not want any interferent to happen, in that case you collect a blood sample in a tube with sodium fluoride and potassium dichromate. Okay, so can we start learning about all blood collection tubes? Do you want to learn? If you want to learn, can I see a thumbs up on the chat box, please? Live chat. So tell me, what color cap does it have? Red color cap. What is the mnemonic for red color cap? It is RSS. Thanks all of you. I'm happy to see the interests. Okay. So thanks. Yeah. What is the mnemonic for red top tube? It is RSS. What does RSS mean? R stands for red. S stands for silica, which is a clot activator. The next S stands for serum. Right. So red, serum, silica. What is silica? It's a clot activator. Clot happens quickly. Serum gets separated. This is used for biochemistry. Whenever you're sending a blood sample to biochemistry laboratory, collect blood sample in a red top tube. What do you estimate in biochemistry? Renal function test, urea, creatinine. Electrolyte estimation, right? Electrolyte estimation. Liver function test. Cardiac function test. Hormone estimation, right? So for all these conditions, you are supposed to collect blood sample in a red top tube. Biochemistry is red top tube. It is RSS. Are you clear? Now, gray. What did I tell you about gray top tube, all of you? Please start answering. G for G. Gray top tube is for glucose estimation. What does it have? It has sodium fluoride and potassium oxalate. Right, that's got sodium fluoride and potassium oxalate. Sodium fluoride inhibits enolase. And now you know it is also used for alcohol estimation. Okay, G for G. Good. Have you seen this? Lavender, right? So lavender top tube has got K2 EDTA or K3 EDTA. Please remember, it can have either K2 EDTA or K3 EDTA, and it is used for pathology. What do I mean by pathology? For complete blood count. In biochemistry also, we expect blood sample to be collected in a lavender top tube if you want HbA1c to be estimated. Right? What if you collect in a red top tube and send it asking for HbA1c? I cannot do that. Because in a red top tube, the clot activator will allow clot to get stabilized. In the clot, RBC will be trapped. If RBC is trapped, I cannot get hemoglobin out of RBCs. That is why for HbA1c estimation, it has to be a lavender top tube which has K2 EDTA or K3 EDTA. Or whenever you have to estimate labile parameters like parathormone, ACTH, ammonia. Labile parameters like parathormone, ACTH and ammonia again collect blood sample in a lavender top tube. So all of you please repeat this. When will you collect blood sample in a lavender top tube? What does it have? It has K2 EDTA or K3 EDTA. It is used for complete blood count, HbA1c estimation, parathormone, ACTH and ammonia. Okay. Labile parameter meaning those which get degraded. Okay, parathormone, ACTH, ammonia levels get affected after sample collection because proteases are present in the sample. These proteases degrade PTH, they degrade ACTH, causing false low values. I don't want to get false low values, so I have to inhibit proteases. How can I inhibit protease? By chelating calcium. What does EDTA do? EDTA chelates calcium. In the absence of calcium, proteases are not active in the sample. These proteases would otherwise degrade PTH, ACTH. Do you understand this? So whenever you're collecting blood samples, say in a chronic kidney disease patient, you want to estimate PTH. Do not collect in a red top tube, collect in a lavender top tube. Are you clear, everybody? Yeah, I will tell you the order of draw also. Okay. Now what is this? Light blue color, right? Light blue color is citrated tube. 3.2% sodium citrate. Light blue color is 3.2% sodium citrate. C for C, it is used for coagulation assays. Sodium citrate is used for coagulation assays. What do you mean by coagulation assays? To estimate prothrombin time, activated partial thromboplastin time, to estimate fibrinogen, to estimate factor assays. For all these, you will have to use a light blue top tube, which is 3.2% sodium citrate tube. Okay. What if it's 3.8% sodium citrate, all of you? 3.8% sodium citrate is a black topped tube. And this black top tube is used for ESR. 3.8% sodium citrate is a black top tube that is used for ESR. Very good. Emergency, it is not blue. Emergency is green. Okay. So do you see a green top tube here? What does it have? Green top tube has got heparin. We have both sodium heparin and lithium heparin. Both are available. And what will you use this heparinized tube for? This is used for emergency or stat sample collection. Okay, wherever you have to estimate something quickly. For example, there is a critically ill patient and you want electrolytes to be estimated. In that case, if you collect blood sample in a red top tube, which I showed you first, you will have to wait for the clot to get stabilized and then you will have to estimate it. That will take a longer period, right? Until then the patient cannot be managed. So whenever emergency or stat sample collection is necessary. For example, electrolyte estimation in a critically ill patient, or troponin estimation in a patient with suspected myocardial infarction. You have to collect blood sample in a heparinized tube. Another application of heparinized tube is for genetics, right? Whenever DNA or RNA has to be extracted, whenever DNA or RNA has to be extracted, again you will have to collect blood sample in a green top tube. So molecular diagnostics and cytogenetics. For both molecular diagnostics and cytogenetics, you collect blood sample in a green top tube. So ABG is no. Okay. Heparinized tube is not for ABG. I keep saying about this. Don't you all remember collecting blood sample in a syringe for arterial blood gas analysis? This is not for ABG. Please remember, for ABG collection, it has to be a syringe based collection. This is a tube. You cannot use this for ABG. Clear? Yes, heparinized syringe. And this is the last tube, right? So what do you see in this tube? Do you see a gel in the bottom? Yeah, you see a gel in the bottom and it is sprayed with clot activator. So what do you call this tube as? You call this tube as gel and clot activator tube. Right, this is called as gel and clot activator tube. So all of you tell me, what is the difference between the red top tube and the yellow top tube? Red top tube has only clot activator sprayed, whereas yellow top tube has got gel and a clot activator that is sprayed. Now what do we do using this tube? In this tube you collect the blood sample. When you collect the blood sample, initially the blood sample will stay on the top of the gel. When you first collect, the blood sample will stay on the top of the gel. Then when you centrifuge, the gel ascends. When you centrifuge, the gel ascends and it stays in between cells in the bottom and serum on top. Do you understand this? After centrifugation, what happens to the gel? The gel ascends up and it stays as a mechanical barrier between cells in the bottom and serum on top, so that even if cells undergo lysis, all of you know after sample collection, in due course a sample will undergo lysis. So even when the sample undergoes lysis, it is not going to contaminate the serum. So what is the function of the gel here? The gel acts like a mechanical barrier. It does not allow the cell lysis to contaminate the serum. So when will you use a gel in a tube? Whenever the sample has to be transported for a prolonged period of time, or whenever the sample has to be stored for a prolonged period of time, you will have to collect blood sample in an yellow top tube. For example, all private laboratories which offer home collection, right? So the phlebotomist goes to the home, collects blood sample. Only towards the end of the day, the sample reaches the laboratory where an analysis is done. Or private laboratories wherein there are satellite collection centers wherein sample collection is done and then it's sent to the main laboratory totally in a different city, right? Again, the transportation takes a longer time. So whenever the sample has to be transported or whenever the sample has to be stored for a prolonged period of time, in biochemistry, when I say biochemistry, for renal function test, electrolyte, liver function test, cardiac function test, hormones. So for all these, you collect blood sample in a yellow top tube when the sample has to be stored or transported. Are you clear, everybody? I hope it's clear. Yeah, so this is about the various types of blood collection tubes and their uses. Okay, so quick recap. Red top tube, RSS. Red has got silica. It is for serum separation. It is for biochemistry. Similar to red top tube is yellow top tube, which has gel in addition. For biochemistry, whenever the sample has to be transported or stored, we use yellow top tube. Okay. Then heparin. Why am I telling you about heparin now? Heparin is also used for biochemistry whenever rapid analysis has to be done. Emergency sample collection, collect it in a green top tube because I don't have to wait for the clot to get stabilized. Immediately I can centrifuge, immediately I can analyze and give the report. So when you become a resident, if you want emergency reports to be released, it is not enough if you just write it in the request form that it is emergency. Do you understand this? It's not enough you just write emergency on the request. You should collect it in a green top tube. Only then we will be able to release the report quickly. So that's about red, yellow and green. Okay. Then I told you about gray. Gray is for glucose estimation. It has got sodium fluoride and potassium oxalate. It is also used for alcohol estimation. Then I told you about lavender. Lavender has K2 EDTA or K3 EDTA used for complete blood count, HbA1c, parathormone, ACTH and ammonia. That's about lavender. Then blue. Blue is citrated, 3.2% sodium citrate for coagulation assays. 3.8% sodium citrate is for ESR, that is black topped tube. Okay, that's all about the various types of blood collection tubes. And then someone was asking me about the order of draw, right? So let me tell you about the order of draw. Why is order of draw important? Order of draw is important because we are now doing a closed collection system. Right? What do you mean by closed collection system? Here we don't use a syringe for sample collection. We use a needle for sample collection. How do we use a needle? One end of the needle will be into the vein of the patient. The other end of the needle will be piercing the tube. Right? It will be piercing the tube. Automatically sample gets collected. Once the sample collection, one tube is over, we remove the tube, we attach another tube. We remove the tube, we attach the next tube. We call it as multi-sampler. Right? Why do we call it as multi-sampler? Because one prick. I am pricking the patient only once, and on the other end of the needle, I am inserting tubes one by one. I am collecting multiple samples in multiple tubes. That is why it is called as multi-sample collection. So in this case, do you see that the needle is piercing the rubber stopper of multiple tubes? So there is always a possibility of contamination that is happening. So what should be drawn first? What should be drawn last? The first one you should draw if the sample is more prone to get affected by contamination. If any form of contamination is going to affect the result, right, that sample has to be collected first. And that is why what should be collected first? Culture. Whenever you collect blood sample for culture in a culture bottle or in a culture tube, that has to be collected first. Otherwise, if the needle is going to pierce all other rubber stoppers and then it is piercing the culture tube, that is going to give false positive reports. So culture sample has to be collected first. Number one. Number two, citrated tubes or the blue top tube, because coagulation assays always get affected whenever there is a contamination. So next is blue top tube or citrated tube for coagulation. Okay, that is followed by the traffic light. What is the sequence that you see in traffic light? Topmost light is red, followed by yellow, followed by green. Right? Red, yellow, green. So serum separated tube or the clot activated tube has to be collected first, followed by gel, and then that is followed by heparin. Okay. Suppose a patient wants tumor markers. Yeah. The patient wants at the same time renal function test, liver function test, tumor marker and molecular diagnostics. All these have to be done. Tumor markers are not usually done in peripheral labs. Tumor markers are done only in reference lab. So this person knows that I have to collect the sample. One sample has to be sent to my lab. The other sample has to be sent to the reference lab. So whenever I have to send it to the reference lab, I am going to collect it in an yellow tube. And if I am going to analyze something in my own lab, let me collect it in red top tube. So for the same patient, you collect blood sample in red top tube first, followed by yellow top tube and then green. Okay. And then that is followed by EDTA or lavender. Do you understand this? That's followed by EDTA or lavender. Finally, it has to be fluoride. Okay, because fluoride is so notorious to cause contamination. So fluoride has to be collected at last. Are you clear about the sequence? right? Which one should be collected first? Obviously culture, followed by coagulation, followed by red, yellow, green, and then it is lavender. Finally, it is fluoride. So now it is part 2. Part 1 is over. Let's proceed to part 2, chemistry. This is an FMGE Jan 2023 MCQ. Which of the following is cardioprotective? What should be your answer? Omega-3 fatty acids. Good. Excellent. Omega-3 fatty acids are more cardioprotective because omega-3 fatty acids reduce triglyceride levels. How do they reduce triglyceride levels? They suppress VLDL synthesis in the liver. Okay, so which is more cardioprotective? Omega-3 fatty acids. Which of the following amino acids primarily act as a buffer in blood due to its ability to accept and donate protons at physiological pH? Please answer. Which amino acid acts as a physiological buffer? It is histidine. The answer is histidine. Okay. So now I am going to give you few one-liners and few mnemonics related to amino acid chemistry. Okay. If you know these facts, any question on amino acid chemistry, you will be able to answer. Okay. So one-liners related to amino acids. The first one-liner is about the simplest amino acid, which is glycine. The simplest amino acid, glycine, does not even have an asymmetric carbon atom. Yeah, it does not even have an asymmetric carbon atom. So it cannot exhibit stereoisomerism. Yeah, glycine, the simplest amino acid, has no asymmetric carbon atom. It cannot exhibit stereoisomerism. It is optically inactive. So if you are asked which is the optically inactive amino acid, all of you give the right answer. It is glycine. That is point number 1. Point number 2, aromatic amino acids. What are the 3 aromatic amino acids? Phenylalanine, tyrosine, tryptophan. Right? Phenylalanine, tyrosine, tryptophan, all 3 aromatic amino acids absorb UV light at 280 nanometer. So what is the second one-liner? Phenylalanine, tyrosine, tryptophan absorb UV light at 280 nanometer. Please memorize the 3 aromatic amino acids. What are the 3 aromatic amino acids? Phenylalanine, tyrosine and tryptophan. Okay. And the third fact, histidine, pK of histidine is 6, which is close to the physiological pH of 7.4. Hence, histidine acts as a physiological buffer. Okay, pK of histidine is 6, which is close to the physiological pH of 7.4. So, histidine acts as a physiological buffer. Okay, the next fact is proline. Proline is an amino acid. It cannot take part in hydrogen bonding. So, proline disrupts alpha helix. So, what are the 2 amino acids which disrupt alpha helix? One is proline, the other one is glycine. Okay, proline and glycine disrupt alpha helix. Okay, so these are the one-liners that you need to know about amino acids. Can you all repeat this? What are the one-liners that I've told you? Please look at the slide that you see here, because as I always tell you, there is no shortcut to memory. It is just repetition. So what are the one-liners? Glycine has no asymmetric carbon atom, no stereoisomerism, optically inactive. Phenylalanine, tyrosine, tryptophan absorb UV light at 280 nanometer. And then proline and glycine disrupt alpha helix. Histidine acts as a physiological buffer. Okay. Now mnemonics. Mnemonics related to amino acids. I am going to tell you 4 mnemonics. The first mnemonic is HIP. Okay. The first one is HIP. The second one is TIA. Okay, the second one is TIA. Have you heard of transient ischemic attack? That's the second mnemonic that you can remember. And the third one is AGS. Okay, AGS. The last one is MPS. AGS and the last one is MPS. These are the 4 mnemonics. So what does HIP stand for? H stands for histidine. I stands for imidazole group. Okay, histidine has which group, all of you? Imidazole group. And it answers Pauli's test. Please, all of you repeat this. They can ask you any of these facts. They can ask you which group is present in histidine. Which color reaction does histidine answer? So HIP, what does HIP stand for? Histidine has imidazole group and it answers Pauli's test. Second one is TIA. T stands for tryptophan. Tryptophan has indole ring. Okay, tryptophan has indole ring and it answers aldehyde test. Okay, it answers aldehyde test. So that's about TIA. AGS, arginine has guanidium group. Okay, arginine has guanidium group and it answers Sakaguchi test. And it answers Sakaguchi test. The last one is MPS. Okay, these are the color reactions which give you red color. MPS are color reactions which give you red color. M stands for Millen's test. Okay, M stands for Millen's test. P stands for Pauli's test. Did I tell you about Pauli's test? S stands for Sakaguchi test. So all these three give you what color? They give you red color reactions. Clear? So tell me, what are the 4 mnemonics? All of you, please repeat it. If you like this mnemonic, please like this video. Okay. So tell me HIP. What does HIP stand for? Histidine, imidazole group, Pauli's test. TIA, tryptophan, indole ring, aldehyde test. AGS, arginine, guanidium group, Sakaguchi test. MPS, Millen's test, Pauli's test and Sakaguchi test. These three give you red color reaction. Okay. Good. Now, which of the following is deficient in the mucopolysaccharidoses associated with Nuttowick's disease? Okay, this is quite strange, right? Usually they ask you about hunters or hurlers, right? But this time they asked you about Nuttowick's disease. Nuttowick's is mucopolysaccharidosis type 9. Okay, there are 7 types of mucopolysaccharidosis, 1 till 9, except 5 and 8. Okay, 7 types, type 1 till type 9, except 5 and 8. And this is mucopolysaccharidosis type 9, caused by the defect of hyaluronidase. If hyaluronidase is defective, which mucopolysaccharide would accumulate? Hyaluronic acid would accumulate. Okay, so can we learn about all these mucopolysaccharidoses? If they ask you about type 9, they can ask you about any other mucopolysaccharidosis, right? So tell me, how many types did I tell you? I said 7 types. What are the 7 types? Type 1 till type 9 except 5 and 8. Okay. Now what is type 1? This all of you know. Please answer. Type 1 is Hurler's syndrome. Type 2 is Hunter's syndrome. Okay, type 1 is Hurler's, type 2 is Hunter's. Type 3, right? This is 3 and S. 3 and S look similar, right? Type 3 is Sanfilippo disease. Okay, type 3 is Sanfilippo's disease, A till D. There are 4 subtypes. Just remember, type 3, 3 and S look similar. Type 3 is Sanfilippo disease. Okay, type 4, 4 is Marque. Okay, Marque A and Marque B disease. Okay, and then type 6. Have you heard of Marque-Lamy disease? Type 6 is Marque-Lamy disease. Type 7. 7 starts with S. Type 7 is Sly disease. Okay, type 7 is Sly disease. Type 9. 9 starts with N. It is Nuttowick's disease. Okay, so these are the names. So can you look at this and memorize it first? What is type 1? I told you the most popular, Hurler's disease. Type 2 is Hunter's disease. 3 and S look similar. 3 is Sanfilippo disease, A to D. Type 4 is Marque A and B disease. Type 6 is Marque-Lamy disease. Type 7 S Sly disease. Type 9 N Nuttowick's disease. Okay, so these are the names. Okay, now enzyme defect. Hurlers is because of the defect of, so type 1, right? What is 1? Roman letter 1 looks like the alphabet I. Roman letter 1 looks like the alphabet I. I stands for iduronidase. Easy to remember, right? Type 1, Roman letter 1, alphabet I stands for iduronidase defect. Type 2 hunters is because of iduronate sulfatase defect. Right? Iduronate sulfatase defect. And iduronic acid is present in heparin sulfate and dermatan sulfate. Okay, iduronic acid is present in heparin sulfate and dermatan sulfate. How do we remember this? I'll give you a shortcut to remember this. I hope you would have all injected heparin to one or the other patient. Right? Heparin you would have injected to one or the other patient. Heparin has to be injected subcutaneously. Right? Heparin has to be injected subcutaneously. One who injects heparin intradermally is an idiot. Do you understand this? One who injects heparin intradermally is an idiot. Just to remember, no offenses. So you can use this statement to remember that iduronic acid is present in heparin sulfate and dermatan sulfate. And you know, in type 1 and type 2, it is iduronic acid metabolism defect. So what are the mucopolysaccharides that accumulate in these two conditions? Heparin sulfate and dermatan sulfate. Are you clear? In both these conditions, heparin sulfate and dermatan sulfate accumulate. Okay. Sanfilippo A to D. In Sanfilippo A to D, the mucopolysaccharide that accumulates is heparin sulfate. Don't worry about the enzyme defect. A to D, all these have different enzyme names. They are not going to ask you that. They might ask you, if at all, they might ask you which mucopolysaccharide accumulates in Sanfilippo A to D. In all these conditions, it's heparin sulfate that accumulates. Okay. Mark you A and B. Mark you A is because of a defect of galactoside sulfatase and galactosidase. Okay, let me write it down here because there is not enough space. Mark you A. Mark you A is because of galactosyl sulfatase defect, whereas mark you B is because of beta galactosidase defect. Okay, both are related to galactose metabolism. Okay, and the one mucopolysaccharide which has galactose. Okay, only one mucopolysaccharide has galactose. Otherwise, mucopolysaccharides have uronic acid and amino sugar. So the only one which has got galactose is keratin sulfate. So in MARQ A and B disease, which mucopolysaccharide accumulates? Keratin sulfate accumulates. Okay. Now Martu-Lamy disease. In the case of Martu-Lamy disease, I want you to just remember this. Okay. Martu-Lamy aryl. Okay. Mort aryl sulfatase R. Okay. Aryl sulfatase defect. And the mucopolysaccharide that is defective in this condition is dermatan sulfate. Okay, Martu-Lamy disease, it is aryl sulfatase defect, and the mucopolysaccharide that accumulates in this condition is dermatan sulfate. Slice syndrome is because of the defect of beta glucuronidase. Okay, it is because of the defect of beta glucuronidase. Additionally, hunters and hurlers, there is accumulation of heparin sulfate and dermatan sulfate. In the case of slice syndrome, there is additionally chondroitin sulfate that accumulates. Okay, Natowitz disease. So what did I tell you? Hyaluronidase defect. In this case, what accumulates? Hyaluronic acid accumulates. Okay. So quick recap on all these mucopolysaccaridosis. At the end of the session, PDF will be uploaded. I want you to watch the slide, memorize it. Just have this in your memory. Nothing else will be asked. If you know this, that's more than enough. So mucopolysaccharidosis, just remember 9 types. 7 types. Type 1 till type 9 except 5 and 8. 1 and 2 we know hurlers and hunters. Iduronidase defect and iduronate sulfatase defect. And we know iduronic acid is present in heparin sulfate and dermatan sulfate. So in 1 and 2, heparin sulfate and dermatan sulfate accumulate. And then I told you 3, Sanfilippo A to D, individual enzymes are defective, but Sanfilippo is characterized by the accumulation of heparin sulfate. Okay, mark you A and B. It is galactose sulfatase and beta galactosidase defect. Just remember the only one mucopolysaccharide which has galactose is keratin sulfate. So in mark you A and B, it is keratin sulfate that accumulates. Okay. Type 6 is Martu-Lamy disease, MAR, right? Aryl sulfatase defect. And then it is dermatan sulfate that accumulates. Type 7, it is Slice syndrome, S7 Slice syndrome. And in Slice syndrome, the enzyme that is defective is beta glucuronidase. Additionally, chondroitin sulfate also accumulates. Type 9, Natovich's disease. And the enzyme that is defective is hyaluronidase, hyaluronic acid accumulates. Difference in hurlers and hunters. Yes. Yes, in main notes also I have not told because I didn't expect them to ask you this question because they have started asking you. I have started discussing it. And please, yes, memorize this table. Just this table is enough. And for those of you who want to know the differences between hunters and hurlers, okay, hunters and hurlers. The first difference is, as I told you, hunters is type 2, hurlers is type 1. We know the enzyme that is defective. In type 1 is just iduronidase. Here it is iduronate sulfatase defect. And I want you to know that hurlers is severe, hunters is relatively milder. Okay, hurlers is severe, hunters is relatively milder. And in hunters, there is no corneal clouding. In hunters, there is no corneal clouding. How do I remember this? In hunter, the vision is perfect. Do you understand this? In hunter, the vision has to be perfect. So, no corneal clouding in hunters. Okay. Similarly, no inguinal hernia in hunters. Okay. No inguinal hernia in hunters. And then hunters is X-linked recessive, hurlers is autosomal recessive. Are you clear about these differences? So what are the differences I told you? Hurlers is type 1, hunters is type 2. Hurlers is iduronidase defect, hunters is iduronate sulfatase defect. And then hurlers is severe, hunters is relatively milder. In hunters, there is no corneal clouding, there is no inguinal hernia. Hunters is X-linked recessive, hurlers is autosomal recessive. Okay. So now about enzymes. This is part 3, enzymes. In enzymes, I'm going to tell you about enzyme inhibition, right? Because that is the only type of MCQ that I've seen so far in FMGE. Okay. So let's learn about enzyme inhibition today by discussing this MCQ. So what kind of inhibition does this graph depict? Okay. So when I look at this graph, I see whenever you see a graph-based MCQ, okay, see what is along x-axis and what is along y-axis, okay. In this case, along x-axis, it is 1 by S. Along y-axis, it is 1 by V. So this is called as double reciprocal plot. Why am I calling it as double reciprocal plot? Because both along the x-axis and along the y-axis, there are reciprocals. Along x-axis, it is 1 by S. Along y-axis, it is 1 by V. And then I see two lines. One line they say is uninhibited. The other line is what happens when there is inhibition. Okay. And what I see here is even in the presence of the inhibitor, Km is unaffected. Right? Even in the presence of an inhibitor, Km is the same. Km is unaffected. So all of you look at this. Normal Km. Please look at what I am writing for some time. Normal Km is a feature of non-competitive inhibition. Do you understand this? Normal Km, non-competitive. In non-competitive, do you see normal Km? Yeah, non-competitive is normal Km. So whenever Km is not affected, what kind of inhibition is it? It is non-competitive inhibition. Okay. So I'm going to tell you the intricacies of double reciprocal plot. All of you please watch this carefully. Okay, for the next 5 minutes. In 5 minutes, we are going to learn all about Lineweaver-Burke plot. We are going to learn all about enzyme inhibition. Okay, so watch this carefully. In Lineweaver-Burke plot, what is along x-axis and what is along y-axis? Along x-axis, it is 1 by S. X and S sound similar, right? X and S sound similar. So along x-axis, it is 1 by S. Along y-axis, it is 1 by V. How to remember this? Y and B look similar. Yes or no? Y and B look similar. So along y-axis, it is 1 by V. Okay. And if you draw a graph like this, for any enzyme, you will always get a straight line. Right? For any enzyme, you will get a straight line. That straight line touches the y-axis at 1 by Vmax. Obviously, right? If along the y-axis it is 1 by V, it touches the y-axis at 1 by Vmax. It touches the x-axis at minus 1 by Km. Right? It touches the x-axis at minus 1 by Km. This is all that you need to know about Lineweaver-Burke plot or double reciprocal plot. So can you all repeat this? What is along x-axis? Along x-axis, it is 1 by S. Along y-axis, it is 1 by V. For any enzyme, you get a straight line. That straight line touches the y-axis at 1 by Vmax. It touches the x-axis at minus 1 by Km. Okay, and always in your exam, you will see two lines. One line will say uninhibited enzyme. The other line will say inhibited enzyme. Okay, so you should compare the uninhibited enzyme curve with the inhibited enzyme curve to find out the type of enzyme inhibition. So look at this. This is Lineweaver-Burke plot. Now about the various types of enzyme inhibition. I am going to tell you about 4 types of enzyme inhibition. The first one obviously is competitive inhibition. The second one is uncompetitive inhibition. The third one is mixed inhibition. The last one is non-competitive inhibition. So all of you tell me, what are the 4 types of inhibition? Competitive, uncompetitive, mixed and non-competitive inhibition. Okay, so in the presence of these inhibitors, what happens to Vmax and what happens to Km? First, about competitive inhibition. In the case of competitive inhibition, all that happens is there is an inhibitor. Let me show the inhibitor in red color. Okay, there is an inhibitor which competes with the substrate. It competes with the substrate to go and bind to the enzyme. Okay, there is just a competition between the inhibitor and the substrate. If it's only a competition, then what happens when you increase a substrate concentration? When you increase the substrate concentration, the substrate will win the competition. The substrate will go and bind. So the take-home message for all of you about competitive inhibitor is, in the case of competitive inhibition, increasing the substrate concentration can overcome the inhibition. Okay, if you can overcome the inhibition, the final Vmax that is achieved is unaltered. Do you understand this? The final Vmax that is achieved is unaltered. So in the case of competitive inhibition, Vmax is normal. Please remember, you can overcome the inhibition by a competitive inhibitor. So Vmax remains normal. Okay, but to reach that Vmax, you have to increase the substrate concentration. So Km is high. Because you have to increase the substrate concentration, Km is high. So all of you tell me, what is the effect of a competitive inhibitor? Vmax remains normal, but Km is high. Okay. Other than this type of inhibition, in the case of any other inhibitor, Vmax is going to be low. Are you clear? Other than competitive inhibition, in the case of any other inhibitor, Vmax has to be low because there is an inhibitor. Okay. Now, uncompetitive inhibition, it is parallel shift. What do I mean by parallel shift? Not only Vmax is low, Km is also low. Do you understand this? Not only Vmax is low, Km is also low. So for uncompetitive inhibition, remember parallel shift, both Vmax and Km are low. Mixed inhibition. In the case of a mixed inhibitor, both Vmax and Km are altered in the wrong direction. Right? Both Vmax and Km are altered in the wrong direction. I want my enzyme to have high Vmax and to have a low Km. Are you clear? For any enzyme, we need to have a higher Vmax and a lower Km. But what happens in the presence of a mixed inhibitor? In the presence of a mixed inhibitor, Vmax is low but Km is high. Do you understand this? In the presence of a mixed inhibitor, Vmax is low and Km is high. And what did I tell you about non-competitive inhibition? I said non-competitive inhibition is normal Km and Vmax is low. Do you understand this? Non-competitive inhibition, normal Km and Vmax is low. So all of you, can you repeat this? What happens to Vmax and Km in the presence of all these inhibitors? Competitive inhibition, you can overcome the inhibition. So the final Vmax is normal but Km is high. Other than this, in all other inhibition, Vmax is low. Uncompetitive inhibition, both Vmax and Km are low. Mixed inhibition, Vmax is low but Km is high. Non-competitive inhibition, normal Km and Vmax is low. Okay. So all of you, I'm going to show you 4 graphs and you're going to find out what kind of inhibition is it. Can you do that? Yeah. So this is along x-axis, you have 1 by substrate concentration. Along y-axis, it is 1 by V. This is the line which is uninhibited. This is the line in the presence of inhibition. Okay. Both uninhibited line and inhibited line touch the y-axis at 1 by Vmax. So 1 by Vmax is unaltered. Vmax is normal. The moment you see Vmax to be normal, what is your answer? What kind of inhibition is it? It is competitive inhibition. Okay. Tell me what happens to Km. This is minus 1 by Km of a normal enzyme. This minus 1 by Km in the presence of an inhibitor. 1 by Km is reduced. So Km is increased. Do you understand this? 1 by Km is reduced, so Km is increased. Whenever Vmax is normal and Km is high, what kind of inhibition is it? It is competitive inhibition. Okay, good. Look at this and tell me. Along x-axis, along y-axis, you see parallel shift. Right? If you see parallel shift, what kind of inhibition is it? Uncompetitive inhibition. Look at this and tell me. In the presence of no inhibitor, this is the 1 by Vmax. In the presence of the inhibitor, 1 by Vmax is high, which means Vmax is low. Okay. In the absence of inhibitor, this is 1 by Km. In the presence of inhibitor, 1 by Km is low. So Km is high. If Vmax is low and Km is high, both are altered in the wrong direction. Very good, all of you. So what kind of inhibition is it? It is mixed inhibition. Vmax is low, Km is high. So what kind of inhibition is it? It is mixed inhibition. Excellent. Look at this. Km is the same. Whenever Km is normal, what kind of inhibition is it? Non-competitive inhibition. Okay. So I hope it's clear to everybody. If you now have the confidence, all of you listen to me. If you now have the confidence to answer graph-based question on enzyme kinetics, please give a thumbs up, meaning like this video. Okay. Cytochrome P450 is associated with. Cytochrome P450 is associated with NADPH. Okay, it is associated with NADPH. So I am going to tell you all about NADPH now. First, let me tell you the uses of NADPH. Then I will tell you the sources of NADPH. All of you listen to this. Only NADH can enter into electron transport chain. Only NADH can give rise to 2.5 ATPs. NADPH cannot enter into electron transport chain. It can never act as a source of ATP. Are you clear? So what are the uses of NADPH? The first use of NADPH is it is necessary for fat synthesis. The first use is it is necessary for fat synthesis. Second is it is used for regenerating glutathione. It is used for regenerating glutathione, which is an antioxidant mechanism in RBCs. That is the second use of NADPH. Third use of NADPH is it is necessary for cytochrome P450 enzymes. Okay, NADPH is necessary for cytochrome P450 enzymes. The last use is it is necessary for ribonucleotide reductase. Whenever you have to convert RNA to DNA, that is done by ribonucleotide reductase enzyme, that needs NADPH. So all of you look at this slide and repeat. What are the uses of NADPH, everybody? NADPH is necessary for fat synthesis. It is necessary for regenerating glutathione in RBCs. It is necessary for cytochrome P450 enzymes. It is necessary for ribonucleotide reductase. Okay. Now, what are the sources of NADPH? The major source of NADPH is HMP shunt. Please answer, which is the major source of NADPH? HMP shunt. Apart from HMP shunt, the second one is cytoplasmic isocitrate dehydrogenase. Usually you learn isocitrate dehydrogenase in citric acid cycle. That is the mitochondrial isocitrate dehydrogenase. That mitochondrial isocitrate dehydrogenase uses NAD and it forms NADH. But there is an isocitrate dehydrogenase that is present in cytoplasm that uses NADP and it forms NADPH. That is the second source of NADPH. The third source of NADPH is malic enzyme. The third source is malic enzyme, which converts malate to pyruvate. So all of you tell me, what are the sources of NADPH? The major source of NADPH is HMP shunt. Minor sources include cytoplasmic isocitrate dehydrogenase and malic enzyme. Okay, so that's about uses of NADPH and sources of NADPH. Please memorize it. Okay. So now part 4, we are done with cell. We then learned about chemistry. We then learned about enzymes. Now metabolism introduction. Okay. So this is an FMGE Jan 2022 MCQ. I hope you have all seen this MCQ. A person gives blood sample for fasting plasma glucose estimation at 7 a.m. 7 a.m. blood sample is given. His last meal was a dinner at 8 p.m. the previous night. Okay. So between 8 p.m. the previous night and 7 a.m. the next morning, how many hours have passed by? 11 hours, right? So 11 hours of fasting. Close to 11 hours of fasting, they're asking you what is the major source of plasma glucose. Please tell me what will be the major source of plasma glucose till 12 to 18 hours. Till 12 to 18 hours after the previous meal, the major source of plasma glucose will be liver glycogenolysis. It is not gluconeogenesis medico-traveler, it is liver glycogenolysis. So the answer is choice B. Okay, but what are we going to discuss now? We are going to now discuss about effects of hormones in the well-fed state and effects of hormones in starvation. Can we do that? Because there are often MCQs asked about it. Effects of hormones in the well-fed state. You're going to answer this. In the well-fed state, tell me what happens to plasma glucose. Plasma glucose is high. When plasma glucose is high, which hormone will be released into the circulation? Insulin will be released into the circulation. And you know, insulin is an anabolic hormone. Right? Insulin is an anabolic hormone. When that is released into circulation, it stimulates all anabolic pathways and it inhibits all catabolic pathways. So what are the anabolic pathways stimulated by insulin? Insulin stimulates glycolysis. Insulin stimulates glycogen synthesis. All of you, please start asking why whenever I tell you something. Why do you think insulin stimulates glycolysis and glycogen synthesis? Thereby, insulin can reduce the blood glucose level. I hope you all know that the major function of insulin is to reduce the blood glucose level. How does it reduce the blood glucose level? By stimulating glycolysis, glucose is converted to pyruvate. By stimulating glycogen synthesis, glucose is converted to glycogen. So it stimulates glycolysis, it stimulates glycogen synthesis, it stimulates fatty acid, cholesterol, triacylglycerol synthesis, it stimulates protein synthesis. Okay, all these are happening in the well-fed state. Simultaneously, it inhibits all catabolic pathways. What are the catabolic pathways it would inhibit? It inhibits glycogenolysis. It inhibits gluconeogenesis. Why do you think it inhibits glycogenolysis and gluconeogenesis? Otherwise, blood glucose level will further increase. So it inhibits glycogenolysis, it inhibits gluconeogenesis. It inhibits peripheral lipolysis. Right? It inhibits peripheral lipolysis. It inhibits fatty acid oxidation. It inhibits protein breakdown. It inhibits amino acid oxidation. So all catabolic pathways are inhibited by insulin. Are you clear about this? Yeah. So this is what is happening in the well-fed state. In the well-fed state, insulin is released. Insulin stimulates all anabolic pathways. It inhibits all catabolic pathways. Now what happens in starvation? This is very important. Right? In starvation, what happens to blood glucose level? It decreases. When glucose level decreases, no insulin release happens. Instead of insulin secretion, all counter-regulatory hormones will be released. So what are the counter-regulatory hormones that are released? All of you listen to this. The first counter-regulatory hormone to be released is glucagon, followed by growth hormone, followed by norepinephrine and cortisol. So tell me, what are the catabolic hormones? Glucagon, growth hormone, norepinephrine, cortisol. And all these catabolic hormones, they are all counter-regulatory to insulin. They are all catabolic hormones. So their main aim is to maintain the blood glucose level. They forget about everything else. They are least bothered about anything else. Their only aim is to maintain blood glucose, because principal cells of your body, namely neurons and RBCs, are dependent only on glucose. I hope you all know that. Yeah, principal cells of your body, namely neurons and glucose, are dependent neurons and RBCs are dependent only on glucose. So when you are in starvation, your body forgets about everything else. It has got only one aim, that is to maintain blood glucose. Okay, to maintain blood glucose, generally we have three ways. The first way obviously is dietary glucose. For you to maintain blood glucose, the first way is dietary glucose. Second way is liver glycogenolysis. The third one is gluconeogenesis. There is no other pathway that can maintain blood glucose. There are three ways by which we maintain blood glucose. One is dietary glucose. Second is liver glycogenolysis. Third is gluconeogenesis. The first one, dietary glucose, can maintain blood glucose only till the first 2 to 2.5 hours. Right? Suppose you had your dinner at 2 p.m. today. Right? Dinner at 2 p.m. Till 4 to 4:30, your dietary glucose will be maintaining the plasma glucose. After 4 to 4:30, contribution from dietary glucose to maintain plasma glucose decreases. But you cannot afford to reduce your blood glucose. So liver glycogenolysis is switched on. So all of you tell me, when is liver glycogenolysis switched on? Liver glycogenolysis switched on 2 to 2.5 hours after the previous meal. So only close to 4 to 4:30 liver glycogenolysis is initiated. And it can maintain blood glucose only till 12 to 18 hours of starvation. Right? Because at that point, all liver glycogen would have got used up. After which liver glycogenolysis cannot maintain blood glucose. That is when gluconeogenesis supports. Okay, that does not mean gluconeogenesis will be initiated only after 12 to 18 hours. There is always an overlap. So when does gluconeogenesis begin? Gluconeogenesis begins 6 hours after the previous meal. Though it starts 6 hours after the previous meal, till the first 12 to 18 hours, what will act as the major source of plasma glucose? 12 to 18 hours. Only after 12 to 18 hours, gluconeogenesis becomes the principal source of glucose. Are you clear about the sequence? Yeah, for the first 2 to 2.5 hours, it's dietary glucose. Then till 12 to 18 hours, it's liver glycogenolysis. At 6 hours, gluconeogenesis is initiated, but only after 12 to 18 hours, it becomes a major source of plasma glucose. Okay. So what is the right answer here? The right answer here is liver glycogenolysis, because it is within 12 to 18 hours. Cortisol not just in the morning hours. Okay, of course there is always a peak in the morning, but glucagon, growth hormone, in that order, the level gets elevated post 2 to 2.5 hours. Do you understand this? It's not like only in the early morning. I said cortisol is the last one to get elevated. Close to 12 to 18 hours is when cortisol release happens. And that you now know corresponds to like previous day, early morning. Okay, so 12 to 18 hours is when cortisol level rise is significant. So what is the right answer here? During starvation, which of the following hormones is primarily produced to maintain blood glucose level? A very simple MCQ asked in the latest Jan 2025 question, glucagon. When does beta oxidation contribute to starvation? Beta oxidation does not directly contribute to starvation. So I was telling you gluconeogenesis is initiated and it becomes predominant after 18 hours, right? And gluconeogenesis is an anabolic pathway. Do you see that? Gluconeogenesis. You're synthesizing something, right? So gluconeogenesis is an anabolic pathway which needs energy. So from where do we get this energy for gluconeogenesis? The energy for gluconeogenesis comes from fatty acid oxidation. So fatty acid oxidation is initiated in starvation because it does not directly give rise to glucose. Fatty acid oxidation supports gluconeogenesis by providing energy. Is that clear? So which of the following is excreted in the patient? So we are done with introduction to metabolism. Now part 5, metabolism. Here we will be learning about all metabolism, carbohydrate, lipids, proteins. After this, we will start genetics. Okay. So which of the following is excreted in the urine of patients with pentosuria? It is xylulose, right? The answer is xylulose. Please remember, it is essential pentosuria. Essential because you don't see any other manifestations. The only finding is pentose is found in urine. So urine will answer Benedict's test. Otherwise, you don't see anything. And it is caused by the defect of xylulose reductase. Essential pentosuria is caused by the defect of xylulose reductase, and xylulose will be found in the urine. So today in my Telegram group, some of the students were asking, one student was asking me about GLUT transporters. So let's learn about GLUT transporters today also. Which receptor plays a crucial role in glucose uptake by skeletal muscle? Answer is GLUT4 transporter. Very good. Right, GLUT4 transporter. So please remember this, insulin dependent. Nobody should forget this. All of you please memorize it. Insulin dependent glucose uptake happens in skeletal muscle, cardiac muscle, and adipose tissue. Okay, it happens in skeletal muscle, cardiac muscle, and adipose tissue. By which transporter upregulation? By GLUT4 transporter upregulation. Okay, so what is the right answer here? The right answer is GLUT4. Always memorize, it is insulin dependent. So now I am going to tell you about 5 types of GLUT transporters, GLUT1 till GLUT5. Okay, so GLUT1 and GLUT3 transporters are present in those cells for which glucose is indispensable. Can you tell me what are the cells for which glucose is indispensable? Just now I told you neurons, RBCs are totally dependent on glucose, and placenta, right? In neurons, RBCs and placenta, which transporter do we have? We have GLUT1 and GLUT3 transporter. Okay, which means GLUT1 and GLUT3 transporters exhibit very high affinity for glucose. And neoplastic cells, I hope you all know the preferred fuel for neoplastic cell is also glucose. And your neoplastic cells are super smart. Okay, that is why they decided to use a GLUT transporter with very high affinity for glucose, and that is GLUT1 transporter. So GLUT1 transporter is also present in neoplastic cells. Okay. Now GLUT2 transporter. GLUT2 transporter is present in those cells which are related to, very good, Warburg effect. Excellent. So GLUT2 transporter is present in those cells which are related to glucose balance. So what are the cells which are related to glucose balance? First, glucose has to be absorbed by enterocytes. Then glucose has to be stored by hepatocytes. And then insulin has to be released by pancreatic beta cells. And glucose has to be reabsorbed by proximal convoluted tubular cells. In all these places, you have GLUT2 transporters. So all of you repeat this. GLUT2 is related to glucose balance. What are the cells which are related to glucose balance? Glucose has to be absorbed by enterocytes. It has to be stored by hepatocytes, pancreatic beta cells, and renal tubular cells. In all these places, you have GLUT2 transporters. GLUT4. What did I tell you about GLUT4 transporter? Insulin dependent glucose uptake happens in skeletal muscle, cardiac muscle, and adipose tissue by GLUT4 transporter upregulation. Okay, insulin dependent. So if you want to know how do I remember this list, how do I remember what are the tissues which are dependent on insulin? Just remember, insulin is an anabolic hormone. So insulin stimulates glucose uptake only into those cells when glucose can be used for anabolism. And glucose is taken up by skeletal muscle and cardiac muscle. And in these places, because glucose can be used for glycogen synthesis. In cardiac muscle and adipose tissue, glucose can be used for fat synthesis. Do you understand this? So insulin dependent glucose uptake happens in skeletal muscle, cardiac muscle, and adipose tissue through GLUT4 transporter upregulation. GLUT5 is related to fructose movement, and it is related to fructose absorption along the small intestine, and it is also present in seminal vesicle. GLUT5 is related to fructose movement, so it is present in small intestine when it is related to fructose absorption, and is also related to semen containing fructose. Right? I hope you all know sperm uses fructose as its fuel, and that fructose comes from seminal vesicle, and seminal vesicle ducts have GLUT5 transporter. Okay, so update about the length of the session in the sense, this session will continue till 11 o'clock, if that is what you're asking, till 11 or 11:30. Okay. So what is the right answer? Which receptor plays a crucial role in glucose uptake by skeletal muscle? It is GLUT4 transporter. Okay. The rate limiting enzyme of HMP shunt. What is the rate limiting enzyme of HMP shunt? Glucose 6-phosphate dehydrogenase. Every year from Biochem, we see 15 to 16 MCQs. The latest FMGE exam, I discuss 17 MCQs from biochemistry. When I say biochemistry, not just the pure biochemistry, clinical biochemistry included. 15 to 16. Okay. Glucose 6-phosphate dehydrogenase. Please don't get confused between glucose 6-phosphatase and glucose 6-phosphate dehydrogenase, all of you. Yeah. So glucose 6-phosphatase, I told you, is present in gluconeogenesis. Glucose 6-phosphate dehydrogenase is an enzyme of HMP shunt. Okay. So let's learn about rate limiting enzymes of all carbohydrate metabolism pathways. Okay. So tell me, what is the rate limiting enzyme of glycolysis? The rate limiting enzyme of glycolysis is phosphofructokinase 1. Surgery boy, thanks. Yes, vitamin questions were integrated with PSM. Yes. Citric acid cycle, the rate limiting enzyme is isocitrate dehydrogenase. For glycogen synthesis, obviously it is glycogen synthase. For glycogenolysis, it is glycogen phosphorylase. Okay, for glycogenolysis, it is glycogen phosphorylase. For gluconeogenesis, the rate limiting enzyme of glycolysis is phosphofructokinase 1. So the rate limiting of gluconeogenesis is something which reverses phosphofructokinase 1 step, and that is fructose 1,6-bisphosphatase. Right, that is fructose 1,6-bisphosphatase. HMP shunt, just now I told you, glucose 6-phosphate dehydrogenase. Mama, I don't know what your name is, but you have named yourself Mama. But you will be able to remember if you know overall about the pathway. Right, there should be no doubt about rate limiting enzyme of glycolysis being phosphofructokinase 1. Right, this should be like ingrained in your brain. Phosphofructokinase 1 cannot be seen in any other pathway, right? So what is the rate limiting enzyme of glycolysis? It is phosphofructokinase 1. Similarly, citric acid cycle is isocitrate dehydrogenase. Glycogen synthesis, the name by itself tells you it is glycogen synthase. Glycogenolysis, you're breaking down glycogen. So the rate limiting enzyme has to be glycogen phosphorylase. Gluconeogenesis, reverse phosphofructokinase 1 step by fructose 1,6-bisphosphatase. HMP shunt, the first enzyme of HMP shunt is glucose 6-phosphate dehydrogenase. Okay, so that's about rate limiting enzymes of all these pathways. So answer this please. Rate limiting enzyme of HMP shunt is glucose 6-phosphate dehydrogenase. What about glucose 6-phosphatase? Which pathway is it related to? Gluconeogenesis. Phosphofructokinase 1 is the rate limiting enzyme of glycolysis. Glycogen phosphorylase is the rate limiting enzyme of glycogenolysis. July 2025 MCQ. A breastfed infant presents with lethargy, hepatomegaly and cataract. Which of the following enzyme deficiencies is most likely responsible for this presentation? So what will be your answer? This is classical galactosemia. Why am I calling it as classical galactosemia? Because the child is mother fed and the child presents with cataract. That is why I am going to call it as classical galactosemia. So the answer is Galput deficiency. Do you understand this? The answer is Galput deficiency. So I am going to name all the conditions related to all four choices here. Galactokinase defect is also called as galactosemia. Okay, galactokinase defect is also called as galactosemia. But in this galactosemia, you will see only cataract. you will not see hepatomegaly. Only in classical galactosemia, what will you see? You will see cataract and hepatomegaly. Aldolase B defect is called as hereditary fructose intolerance. Aldolase B defect is called as hereditary fructose intolerance. And what is fructokinase defect? Fructokinase defect is essential fructose urea. It is essential fructose urea. So all of you repeat this. In fructose metabolism disorders, there are two. Galactose metabolism disorders, there are two. Okay, let me first tell you about fructose metabolism disorder. The first enzyme fructokinase. For any hexose, the first enzyme will be a kinase. So the first enzyme fructokinase defect causes essential fructose urea. Whenever you see the term essential, it means you do not see any other defects. Essential meaning the person is absolutely normal. The only finding is fructose is found in urine. That happens when fructokinase is defective. When aldolase B is defective, that is called as hereditary fructose intolerance. In hereditary fructose intolerance, apart from fructose being found in urine, you will see jaundice, you will see hepatomegaly, you will see hypoglycemia as well. Okay. Galactose metabolism disorders are galactosemias. One is just galactosemia, the other one is classical galactosemia. Galactosemia is caused by galactokinase defect. In that case, galactose will be found in urine and there is cataract. Okay. Classical galactosemia, galactose is found in urine, there is cataract. Additionally, you see jaundice, hepatomegaly. Do you understand this? This is how you differentiate. So you start from the history. If the history says mother-fed child, mother's milk has lactose. Lactose gets digested to give galactose and glucose. So if you see the history, even when the child is mother-fed, it is galactose metabolism disorder. It is either galactosemia due to galactokinase defect or it is classical galactosemia due to galput defect. How will you differentiate the two? If there is only cataract, it is galactokinase defect. Right? If there is only cataract, it is galactokinase defect. If there is jaundice, hepatomegaly, hypoglycemia, it is classical galactosemia. Okay. Whereas if the history says the child presents after transition from mother's milk to infant foods. Yeah, if the history says the child presents after transition from mother's milk to infant foods, which means until the child is mother fed, no defect. Which means galactose metabolism is normal. The moment infant food is started, infant foods are rich in sucrose. Sucrose gets digested to give glucose and fructose. After fructose enters, if you see all these manifestations, then it is fructose metabolism disorder. Right? And if there is only fructose in urine, it is essential fructose urea. If you see jaundice, hepatomegaly, then that is hereditary fructose intolerance caused by aldolase B defect. I hope it's clear to everybody. Yes, classical galactosemia or galactosemia always presents with oil drop cataract. If it's clear, please like the video and give a thumbs up. Okay. So it is oil drop cataract that you see in galactose metabolism disorders. Okay. So this is a June 2021 MCQ. FMGE. 8-month-old infant presents with hypoglycemia. So the presenting feature is hypoglycemia. Already liked. Thanks. So 8-month-old infant presents with hypoglycemia. On examination, you find hepatomegaly. Blood investigation revealed lactic acidosis, ketosis, and xanthomas. What is the most probable enzyme deficiency? Right? Whenever you see xanthomas, lactic acidosis, ketosis, hypoglycemia, think of von Gierke's disease. Yeah, what is this? This is von Gierke's disease. caused by the defect of glucose 6-phosphatase. So the answer here is choice D, glucose 6-phosphatase defect. Okay, before I tell you about von Gierke's disease, let me tell you how did I exclude other choices. Branching enzyme defect. What is the function of branching enzyme? It introduces branches in glycogen. Okay, it introduces branches in glycogen during glycogen synthesis. Why do you introduce branches in glycogen? We introduce branches in glycogen to provide compactness to glycogen. So if there is a branching enzyme defect, there is no compactness to glycogen. Glycogen occupies a larger space in the liver. What will happen when glycogen occupies larger space in the liver? That causes hepatomegaly. So what will be the principal finding of branching enzyme defect? Branching enzyme defect will present as hepatomegaly that progresses to liver cirrhosis. They never present with hypoglycemia. That is why I am excluding branching enzyme defect. Why did I exclude glycogen synthase defect? What does glycogen synthase do? It synthesizes glycogen both in liver and in muscle. Do you understand this? It synthesizes glycogen both in liver and in muscle. So when that enzyme is defective, both liver and muscle cannot synthesize glycogen. When liver cannot synthesize glycogen, when there is a fasting state, there will be hypoglycemia. Okay. When muscle does not synthesize glycogen, muscle cannot work out. So it presents with exercise intolerance. Do you understand this? Presents with exercise intolerance. So if it was glycogen synthase defect, it should have presented with both hypoglycemia and exercise intolerance. But I don't see any exercise intolerance here. There is only hypoglycemia. That is why it is not choice B. Okay. What if it's acid maltase defect? Acid maltase is otherwise called as alpha glucosidase. Acid maltase is otherwise called as alpha glucosidase. This enzyme defect is called as Pompey's disease. All of you please memorize it. Acid maltase or alpha glucosidase defect is called as Pompey's disease. In Pompey's disease, the pump is affected. Which is the pump of your body? Heart. So it presents with cardiomegaly. Pompey's disease presents with cardiomegaly, cardiomyopathy. Okay. And they present with hypotonia as well. Cardiomegaly, cardiomyopathy, hypotonia. Do I see any of those manifestations in the history? No. And that is why I've excluded choice C as well. So I end up choosing choice D, which is glucose 6-phosphatase defect, which is von Gierke's disease. So now we are going to learn all about glycogen storage disorders. Can we start doing that? Yeah, glycogen storage disorders. So you will have to learn about 9 types of glycogen storage disorders, type 0 till type 7 and then type 11. Okay, you'll have to learn about 9 types of glycogen storage disorders, 0 to 7 including 11. Okay, so what is type 0? Type 0 is glycogen synthase deficiency. Yeah, type 0 is glycogen synthase deficiency. Now do you understand why it's called as type 0? Because it's a glycogen storage disorder without any glycogen storage. If synthase is defective, it cannot store glycogen in any way. So it's a glycogen storage disorder without any glycogen storage. That is why it's called as type 0. Okay. Type 1, 1 stands for von. 1 stands for von. So what is type 1? Type 1 is von Gierke's disease. Okay, type 1 is von Gierke's disease and it is caused by the defect of which enzyme, all of you? Glucose 6-phosphatase defect. Okay, so please remember glucose 6-phosphatase is an enzyme of not only gluconeogenesis, it is also an enzyme that is necessary for glycogenolysis to increase blood glucose. So whenever there is a defect of glucose 6-phosphatase, neither glycogenolysis nor gluconeogenesis can increase blood glucose. So that causes severe fasting hypoglycemia. Do you understand this? That causes severe fasting hypoglycemia. Anything which is severe will be type 1. Okay, that is why type 1 is von Gierke's. That is why type 1 is von Gierke's disease, which is caused by the defect of glucose 6-phosphatase. Okay. Now, what is type 2? What happened, Prem Kishore? Why such emoticons? What is type 2? Type 2 is Pompey's disease. Okay, type 2 is Pompey's disease. Just now I told you Pompey's disease is caused by the defect of acid maltase or alpha glucosidase. Acid maltase or alpha glucosidase defect. And what did I tell you about Pompey's? It presents with cardiomegaly, cardiomyopathy and hypotonia. Okay. Type 3. All of you tell me which is the third alphabet. Type 3, which is the third alphabet? C. So type 3 is Cory's disease. After C, the alphabet is D. Cory's disease is caused by the defect of D-branching enzyme. Okay, Cory's disease is caused by the defect of D-branching enzyme. So type 3 is C and D. Type 4 is A and B. I already watched your LRR posted 7 months ago. So should I watch it again or I use it as a revision one for last days? So there will be few additions here. If you have time, you can watch this now. I would recommend that all of you watch this session live. Okay, because if you watch the session live, then you have no other go but to follow my speed, right? And I have no other go but to complete the entire biochemistry in the next 2 hours. So if you're watching this live along with me, then you would have finished biochemistry 1 revision in 1 go. Okay, and I will be repeating everything multiple times. So along with me, if you repeat, then you would have memorized it also along with me. So that few days before exam, you can just watch this on a faster pace or faster mode and the entire revision is done. Okay, so do it along with me if that answers your question. So type 4 is A and B. A stands for Anderson's disease. I learn so much live, I get so much sitting too. Yes. So Anderson's disease. Anderson's disease is caused by the defect of branching enzyme. Do you understand this? Type 3 is C and D. Type 4 is A and B. Anderson's disease, branching enzyme defect. Type 5, Roman letter 5 looks like the alphabet V. Right? Roman letter 5 looks like the alphabet V. Can you invert V? Write it twice. Invert V, write it twice, which is alphabet M. So type 5, M stands for McArdle's disease. Okay, McArdle's disease is caused by the defect of M for M. M for M, muscle phosphorylase defect. Type 6 is Her's disease. H for H, hepatic phosphorylase defect. Her's disease is hepatic phosphorylase defect. Type 7 is Tarow's disease. Okay, type 7 is Tarow's disease. It is caused by the defect of phosphofructokinase 1. Did we learn about phosphofructokinase 1, the rate limiting enzyme of glycolysis? And then type 11 is Fanconi-Bickel syndrome. Okay, type 11 is Fanconi-Bickel syndrome. It is caused by the defect of GLUT2 transporter. Are you clear? It is caused by the defect of GLUT2 transporter. So tell me, can you repeat this about all these glycogen storage disorders? Repetition is the only way to memorize. So please repeat it along with me. Type 0, glycogen synthase deficiency, no glycogen storage at all. Type 1, von Gierke's disease, glucose 6-phosphatase defect, 1, von. Type 2, Pompey's disease, defect of acid maltase or alpha glucosidase. Type 3, C and D, Cory's disease, D-branching enzyme defect. Type 4, A and B, Anderson's disease, branching enzyme defect. Type 5, McArdle's disease, M for M, muscle phosphorylase defect. Type 6, Hers disease, hepatic phosphorylase defect. Type 7, Tarow's disease, phosphofructokinase 1 defect. Type 11 is Fanconi-Bickel syndrome. It is caused by the defect of GLUT2 transporters. Okay, so now let's learn about von Gierke's disease. What are the manifestations that you expect here? So I told you von Gierke's disease is because of the defect of glucose 6-phosphatase, in which case glucose 6-phosphate cannot be converted to glucose, right? So that causes severe fasting hypoglycemia. Do you understand this? That causes severe fasting hypoglycemia. Additionally, the accumulated glucose 6-phosphate has no other go but to enter into glycolysis. When glucose 6-phosphate enters into glycolysis, all of you tell me, did I tell you glucose 6-phosphate becomes 2-pyruvate, right? And then I told you these 2-pyruvate become 2-acetyl-CoA. Then I said these 2-acetyl-CoA enter into citric acid cycle and they come out as carbon dioxide. Okay, this is happening normally. So when there is excess glucose 6-phosphate that has accumulated, it enters into glycolysis forming 2-pyruvate. These 2-pyruvate become 2-acetyl-CoA. Okay, there is excess acetyl-CoA that is formed that will be used for fatty acid synthesis and cholesterol synthesis. All of you please remember, acetyl-CoA is a building block for both fatty acid and cholesterol. Because in this condition there is excess acetyl-CoA that is formed, that becomes fatty acid and cholesterol. Excess fatty acid becomes triacylglycerol. Excess cholesterol becomes cholesterol ester. This triacylglycerol accumulates in the cheeks. Okay, it accumulates in the cheeks. So these children have chubby cheeks. Okay, and hence the name doll-like faces. So whenever the history says there is a child presenting with doll-like faces, please remember chubby cheeks because of triglyceride accumulation that is seen in von Gierke's disease. This triglyceride also accumulates in organs, and when it accumulates in liver, it causes hepatomegaly. When it accumulates in kidney, it causes renomegaly. So the child also presents with hepatomegaly and renomegaly. Excess cholesterol causes xanthomas. So these children present with xanthomas everywhere. Okay, not always acetyl-CoA will be used for fatty acid and cholesterol synthesis. Sometimes acetyl-CoA will be condensed to form ketone bodies. Acetyl-CoA condenses to form acetoacetate, which is a ketone body. So these children present with ketosis as well. Not always pyruvate become acetyl-CoA. Pyruvate can also become lactate. So they present with lactic acidosis. Now do you understand why ketosis, why lactic acidosis? Because excess glucose 6-phosphate is entering into all these pathways. Sometimes glucose 6-phosphate enters into HMP shunt. Yeah, because which is the rate limiting enzyme of HMP shunt? Can you all answer? HMP shunt, the rate limiting enzyme is glucose 6-phosphate dehydrogenase. So this glucose 6-phosphate enters into HMP shunt. When it enters into HMP shunt, it forms ribose 5-phosphate. Hence HMP shunt is otherwise called as pentose phosphate pathway. You know that, right? HMP shunt is otherwise called as pentose phosphate pathway because it forms ribose 5-phosphate. This ribose 5-phosphate is used for purine and pyrimidine synthesis. Excess purine will be broken down to form uric acid. Excess purine is broken down to form uric acid. Hence, a child presents with hyperuricemia. Do you understand this? So that is about the manifestations of von Gierke's disease. Till now, I've seen MCQs in FMGE to be focused around von Gierke's disease. So you will have to learn in and out about von Gierke's disease, starting from which enzyme defect causes this condition. All of you please repeat it along with me. Von Gierke's disease is caused by the defect of glucose 6-phosphatase. So it presents with severe fasting hypoglycemia. And along with that, glucose 6-phosphate accumulates. that enters into glycolysis, forms 2-pyruvate. Those 2-pyruvate become 2-acetyl-CoA. Those 2-acetyl-CoA will be used for fatty acid and cholesterol synthesis. Fatty acids become triacyl glycerol. Cholesterol becomes cholesterol ester. So the child does not only present with, all of you answer, the child does not only present with fasting hypoglycemia, the child also presents with hypertriglyceridemia and hypercholesterolemia. Okay, and what are the manifestations of hypertriglyceridemia? The child presents with doll-like faces, with chubby cheeks. The child presents with hepatomegaly. The child presents with renomegaly. Because of hypercholesterolemia, there are xanthomas everywhere. Sometimes acetyl-CoA condenses to form ketone bodies, so you see ketosis. Pyruvate becomes lactate, so you see lactic acidosis. Glucose 6-phosphate enters into HMP shunt, forms ribose 5-phosphate, that forms purine and pyrimidine. Purines on catabolism form uric acid, so you see hyperuricemia. Okay, so these are the biochemical and the clinical features of von Gierke's disease. Are you clear? Yeah. So what is the right answer here? So can you explain everything now? There is hypoglycemia, there is hepatomegaly. Blood investigations reveal lactic acidosis, ketosis, xanthomas. So we know the enzyme defective is glucose 6-phosphatase. Okay. A farmer sprayed a chemical in the pool which causes inhibition of complex 3 of electron transport chain. Identify the inhibitor. Okay, so electron transport chain inhibitor, complex 3 is inhibited by 3-letter word. Complex 3 is inhibited by a 3-letter word which is BAL and antimycin. Okay, BAL and antimycin. So there are 2 right answers here. BAL and antimycin, you can choose one of the 2. I don't think both were given among the choices. It is either BAL or antimycin. Okay, yeah. So can we learn about all electron transport chain inhibitors? Yeah. So to learn about electron transport chain inhibitors, let me tell you the complexes first. And then I will tell you about what happened to surgery boy. Why are you crying or are you laughing? So complexes of electron transport chain. We start from complex 1. Right? What is complex 1? Any break. I thought I'll give you a break after finishing metabolism before genetics. Or if you want break now, probably I'll give you a break after we finish electron transport chain. Is that okay? Yeah. We'll discuss electron transport chain and then you can go for your dinner break. Okay, 5 minutes break. So complexes of electron transport chain, we start from complex 1. Complex 1 is NADH linked dehydrogenase. Complex 2 is succinate dehydrogenase. Okay. Complex 3 is cytochrome B and cytochrome C1. Okay. Complex 3 is cytochrome B and cytochrome C1. Complex 4 is cytochrome A and A3. Finally, complex 5 is ATP synthase complex, which I will not be showing you here. So tell me, what are the 5 stationary complexes? The 5 stationary complexes are complex 1 or NADH linked dehydrogenase, complex 2 or succinate dehydrogenase, complex 3 or cytochrome B and C1, complex 4 cytochrome A and A3, and complex 5 is ATP synthase complex. Apart from these stationary complexes, you also have 2 mobile complexes. One is ubiquinone or Q, placed between 1 and 3. The other one is cytochrome C, placed between 3 and 4. Q between 1 and 3, C between 3 and 4. Okay. Now, what is the direction of transport of electron? From NADH, electrons enter into electron transport chain through complex 1. If it's NADH, it enters into electron transport chain through complex 1. From complex 1, electrons go to Q. Please answer, from Q, where should they go? They go to complex 3. From 3, electrons go to C. From C, electrons go to 4. From 4, the final acceptor of electron is oxygen. This was asked in NEET PG. One of the MCQs in NEET PG is, which is the final acceptor of electron and electron transport chain? Your answer should be oxygen. Okay. Now, if it is FADH2, succinate dehydrogenase FADH2 enters into electron transport chain through complex 2. From complex 2, electrons go to Q. From Q, what is the direction? The same direction, right? Q to 3, 3 to C, C to 4, 4 to oxygen. Okay. Apart from succinate dehydrogenase, you have other FADH2 linked dehydrogenases. Those FADH2 linked dehydrogenases send their electrons directly to Q. Okay. So this is about direction of transport of electron. Now about inhibitors. Inhibitors of electron transport chain. All of you listen to this carefully. This has been frequently asked. Electron transport between complex 1 and Q. Okay, electron transport between complex 1 and Q is inhibited by amobarbital. Okay, the clue is all three names start with A. 1, the first alphabet is A. 1 and Q is inhibited by 3 inhibitors, all of which have something to do with A. The first one is amobarbital, starts with A. The second one is pyrisidin A, ends with A. Pyrisidin A ends with A. The third one is rotenone. Rotenone sounds like vitamin A, right? It is not vitamin A, but sounds like vitamin A. Okay, so rotenone. Otherwise, you can have this, right? Rotenone, what are the last 3 letters? 1. Right? 1, it inhibits between complex 1 and Q. So, electron transport between 1 and Q is inhibited by amobarbital, pyrisidin A and rotenone. Complex 2 or succinate dehydrogenase. Complex 2 or succinate dehydrogenase is inhibited by malonate. Electron transport between 2 and Q. Between 2 and Q is inhibited by, 2 starts with T, right? 2 starts with T. 2 to Q is inhibited by TTFA and carboxin. Between 2 and Q is inhibited by TTFA and carboxin. Between 3 and C is inhibited by a 3-letter word. What did I tell you? 3-letter word is BALN, antimycin. Okay, BALN, antimycin. Here, antimycin is something which is thrown, right? Which is spread on the swimming pool to remove algae. So the answer for that is antimycin, because I forgot that clue that was in the question. A farmer sprayed something on the field, right? So to remove algae or to remove weed, antimycin is thrown. So antimycin is the right answer for that question. So 3-letter word BALN, antimycin. Between 4 and oxygen is inhibited by hydrogen sulfide, cyanide and carbon monoxide. Okay, between 4 and oxygen is inhibited by common poisons, namely hydrogen sulfide, cyanide and carbon monoxide. Are you clear? So what is the list of inhibitors that you know? Between 1 and Q is inhibited by amobarbital, pyrisidin A and rotenone. Complex 2 or succinate dehydrogenase is inhibited by malonate. Between 2 and Q is inhibited by TTFA and carboxin. Between 3 and C is inhibited by a 3-letter word BALN, antimycin. Between 4 and oxygen is inhibited by hydrogen sulfide, cyanide and carbon monoxide. Okay. Oligomycin, one of you have asked about oligomycin. Oligomycin inhibits FO component of ATP synthase complex. I told you ATP synthase is complex 5. So complex 5 is inhibited by oligomycin. Is that clear, medico traveler? So tell me, what is the right answer here? Farmer sprayed a chemical in the pool which causes inhibition of complex 3. So what is the right answer here? The right answer here is antimycin. Does not fit into this, right? We don't throw BAL into the pool. So the answer is D, antimycin. The rate limiting enzyme of cholesterol synthesis is. So can we discuss this and then you can go for a break? Yeah. Rate limiting enzyme of cholesterol synthesis is HMG-CoA reductase. You know statins, right? Statins act by inhibiting HMG-CoA reductase, thereby reducing the blood cholesterol level. So just remember this, cholesterol synthesis, the rate limiting enzyme is HMG-CoA reductase. So can we learn about rate limiting enzymes of all lipid metabolism pathway? We already learnt about carbohydrate metabolism pathway, now about lipid metabolism pathways. Fatty acid synthesis. Did I tell you the precursor of fatty acid is acetyl-CoA? I said that precursor of fatty acid is acetyl-CoA. The rate limiting enzyme is acetyl-CoA carboxylase. I will tell you about uncouplers after you come from the break. Okay. Fatty acid synthesis. What is the rate limiting enzyme? Acetyl-CoA carboxylase. Fatty acid oxidation. I hope you all remember fatty acid oxidation happens in mitochondria. As fatty acids, they cannot enter into mitochondria. They need a carrier. That carrier is carnitine. Okay, that carrier is carnitine. So if they ask you which carrier carries fatty acid into mitochondria, your answer should be carnitine. And if they ask you which is the rate limiting enzyme of fatty acid oxidation, your answer should be carnitine acyl transferase 1. All of you repeat this. The rate limiting enzyme of fatty acid oxidation is carnitine acyl transferase 1. Cholesterol synthesis, just now I told you, HMG-CoA reductase. Okay, statins inhibit HMG-CoA reductase to reduce the blood cholesterol level. So so the rate limiting enzyme of cholesterol synthesis is HMG-CoA reductase. Ketone body synthesis. The rate limiting enzyme of ketone body synthesis is HMG-CoA synthase or HMG-CoA lyase. Please remember the first few steps of cholesterol synthesis and the first few steps of ketone body synthesis are the same. So the rate limiting enzyme are similar. They similarly sound. Okay. So the rate limiting enzyme of cholesterol synthesis is HMG-CoA reductase. For ketone body synthesis, it is HMG-CoA lyase or HMG-CoA synthase. For bile acid synthesis, I told you bile acids are derivatives of cholesterol. Cholesterol undergoes hydroxylation. Cholesterol undergoes hydroxylation to form bile acids. So the rate limiting enzyme of bile acid synthesis is 7-alpha hydroxylase. Okay, the rate limiting enzyme of bile acid synthesis is 7-alpha hydroxylase. So can you all repeat this? Rate limiting enzyme of fatty acid synthesis. Remember the building block of fatty acid is acetyl-CoA. So the rate limiting enzyme is acetyl-CoA carboxylase. Fatty acid oxidation, the carrier is carnitine. So the rate limiting enzyme is carnitine acyl transferase 1. Cholesterol synthesis, HMG-CoA reductase. Ketone body synthesis, HMG-CoA lyase or synthase. Bile acid synthesis, it is 7-alpha hydroxylase. Okay. So, how many of you want to go for... No, you don't have to learn about the whole pathway, Amrita. How many of you want to go for a dinner break? I'm happy that it helped, Alka. I'm happy that the previous LRR session helped you. And this LRR session will also be similar to that. We will cover all topics. Okay. Yeah. So you can go for a quick dinner break and please be back in 10 minutes. So can we start? So a 6 months old infant is brought to the emergency department. Is there anything that I missed? Okay. Brought to the emergency department with lethargy, vomiting, and poor feeding. The episode occurred after prolonged fasting. Lab results reveal hypoglycemia. So the first positive finding that you see here is fasting hypoglycemia, low ketone levels. So this is a classical condition that you should remember, which is non-ketotic fasting hypoglycemia. Whenever you see non-ketotic fasting hypoglycemia, you should think of fatty acid oxidation defect. And if you're asking me why, because I told you fatty acid oxidation is necessary to provide energy for gluconeogenesis. So whenever there is a fatty acid oxidation defect, gluconeogenesis does not get the necessary energy, and that causes hypoglycemia. And usually when there is hypoglycemia, you expect ketosis. But if the hypoglycemia is because of fatty acid oxidation defect, one second. Okay. But if the hypoglycemia is because of fatty acid oxidation defect, it will present as non-ketotic fasting hypoglycemia, because only when there is fatty acid oxidation, you get acetyl-CoA. Only when there is acetyl-CoA, you will be able to form ketone bodies. So fatty acid oxidation defect will present as non-ketotic fasting hypoglycemia. So you know the answer now already. It's a defect of fatty acid oxidation. And if you look at these choices, the right answer is choice B, medium chain acyl-CoA dehydrogenase deficiency. Because what is acyl-CoA? Acyl-CoA meaning fatty acid, dehydrogenation meaning oxidation. So acyl-CoA dehydrogenase is an enzyme of fatty acid oxidation. Medium chain acyl-CoA dehydrogenase defect is fatty acid oxidation defect, and that presents as non-ketotic fasting hypoglycemia. So what is the right answer here? The right answer here is choice B. A man presents with, one second. So a man presents with high VLDL and chylomicrons with eruptive xanthomas. What is the most likely diagnosis? So whenever you see eruptive xanthomas, it means it is hypertriglyceridemia. Tendon xanthomas are different from eruptive xanthomas. Whenever you see eruptive xanthomas, that's a sign of hypertriglyceridemia. So what is the right answer here? The right answer here is choice C, familial hypertriglyceridemia. But today I'm going to tell you how to make a diagnosis given a set of clinical features. Okay. So let's learn about Fredrickson's classification. Fredrickson's classification of hyperlipoproteinemias. How many types do we have? There are 6 types. Right? What are the 6 types? Type 1, 2A, 2B, 3, 4 and 5. So all of you remember Fredrickson's classification 6 types. Type 1, 2A, 2B, 3, 4 and 5. Okay. And for our understanding, I'm going to classify these 6 types into 3 categories. The first category presents with only an elevation of cholesterol. The second category presents with only an elevation of triglyceride. The third category presents with an elevation of both cholesterol and triglyceride. Okay, and the reason behind this classification is because clinical features differ based on this. In a patient, if there is an elevation of only cholesterol, then they present with tendon xanthomas. If there is elevation of only cholesterol, then they present with tendon xanthomas and they present with accelerated atherosclerosis. So what are tendon xanthomas? These are small eruptive lesions which are present along the line of attachment of tendons. Where are tendons more pronounced? Along the knuckles or along the ankles. So if you see small eruptive lesions along the line of attachment of tendons, for example, along the wrist or along the ankle, then that is a sign that your patient has got elevated cholesterol and that person can present with accelerated atherosclerosis. Whereas in your patient, if there is an elevation of triglyceride, then they present with eruptive xanthomas. Elevation of triglyceride, they present with eruptive xanthomas and they present with recurrent pancreatitis. On examination, you find lipemia retinalis. So what are the findings of elevation of triglyceride, eruptive xanthomas, recurrent pancreatitis? On examination, you find lipemia retinalis. Whereas if your patient has got, so if you want to know what are eruptive xanthomas, these are eruptive xanthomas. These are even tinier than tendon xanthomas and they are present along the extensor surfaces of the limb, along the back of elbow. Don't get confused between these eruptive xanthomas and phrenoderma. What is phrenoderma? Phrenoderma is follicular hyperkeratosis caused by vitamin A deficiency or essential fatty acid deficiency. What is phrenoderma? Follicular hyperkeratosis, wherein you will see small papillar lesions like this with a keratin plug at the tip. Do you see white plug at the tip? So whenever you see white plug at the tip, that is phrenoderma or follicular hyperkeratosis. Don't get confused between eruptive xanthomas and phrenoderma. Okay, so if you see eruptive xanthomas, then it's a sign of hypertriglyceridemia. And if your patient has elevation of both cholesterol and triglyceride, then you'll see a combination of both tendon xanthomas and eruptive xanthomas, accelerated atherosclerosis and recurrent pancreatitis. Okay, so if the history says tendon xanthomas, always remember hypercholesterolemia. If the history says eruptive xanthoma, always remember hypertriglyceridemia. Now, out of the 6 types of hyperlipoproteinemias, only one type fits into the first category wherein there is an elevation of only cholesterol, and that is type 2A. Only in type 2A, only cholesterol is elevated. That is why type 2A is otherwise called as familial hypercholesterolemia. Logical name, right? If in this type only cholesterol is elevated, what will you call it as? Familial hypercholesterolemia. And this is caused by a defect of LDL receptor. So tell me, what is expected when LDL receptor is defective? What is the function of LDL receptor? These LDL receptors are present in the liver and they are present in extrahepatic tissues. Their main function is to remove LDL from the blood. And if LDL receptor is defective, LDL will not be removed from the blood, causing elevation of LDL concentration in the blood. LDL carries cholesterol, so blood cholesterol level will be high. Hence the name familial hypercholesterolemia. Okay, and it is an autosomal dominant disorder. That's all about the first category wherein there is an elevation of cholesterol. Are you clear? So it is type 2A familial hypercholesterolemia caused by LDL receptor defect. Now, what are the hyperlipoproteinemias wherein triglyceride levels are elevated? Triglyceride levels are elevated in type 1, type 4 and type 5. Triglyceride levels are elevated in type 1, type 4 and type 5. Easy to remember because you know tri is 3 and 1 plus 3 is 4. Right? Tri is 3, 1 plus 3 is 4. So 1, 4 and 5 are hypertriglyceridemias. Out of these 3 types, type 1 is familial chylomicronemia syndrome. I think somebody asked, Dark Medico asked, is it the same as familial chylomicronemia? Familial chylomicronemia syndrome is type 1. Just remember this. Whenever you learn about lipoprotein, the first lipoprotein that you learn about is chylomicron. So type 1 is related to that chylomicron. It is familial chylomicronemia syndrome. Type 4 and type 5 are otherwise called as familial hypertriglyceridemia. Type 4 and type 5 are otherwise called as familial hypertriglyceridemia. Both are caused by the same genetic defect. And if there is only the genetic defect which causes elevation of triglyceride, it is type 4. If the genetic defect is superimposed by something else which is precipitating further hypertriglyceridemia, that's called as type 5. So when Fredrickson classified, he thought type 4 and type 5 are two distinct disorders. But later we found out that in both type 4 and type 5, there is a common genetic defect which causes an increase in the rate of VLDL synthesis and decrease in the rate of VLDL catabolism, causing an increased VLDL level. VLDL carries triglyceride, hence the name familial hypertriglyceridemia. So in type 4, VLDL level alone is elevated. In type 5, there is a precipitating factor. For example, a type 4 individual who is obese, a type 4 individual who is on oral contraceptive pills, a type 4 individual who is a chronic alcoholic. There is an additional precipitating factor which makes the condition severe. In that case, that is called as type 5. So in type 4, only VLDL levels are high. In type 5, both VLDL and chylomicron levels are high. So tell me, what are the three hyperlipoproteinemias wherein triglyceride level is elevated? Type 1, familial chylomicronemia syndrome. Type 4, type 5, familial hypertriglyceridemias. As the name indicates, in familial chylomicronemia syndrome, chylomicron levels alone are elevated. Type 4 familial hypertriglyceridemia, only VLDL level is elevated. Type 5, both chylomicron and VLDL levels are high. In all these three types, you see eruptive xanthomas, recurrent pancreatitis. On examination, you will find lipemia retinalis. That is about the second category. Now, when do you see an elevation of both cholesterol and triglyceride? That is type 2B and type 3. In type 2B and type 3, both cholesterol and triglyceride levels are high. Out of these two types, you need to know about type 3. Type 3 is ApoE defect. 3 and E sound similar. Type 3 is ApoE defect. In type 3, you see broad beta band. In type 3, you see a broad beta band on lipoprotein electrophoresis. That is why type 3 is otherwise called as broad beta disease. Okay, so that is about the various types of hyperlipoproteinemias. So till now, I've told you this in all my sessions. Till now, I have not seen a conclusive evidence for 6 becoming 7 in sickle cell anemia. So till date, I am confirming it as it is a mutation of the 6th codon. Okay, Anushka. So that is about hyperlipoproteinemias. So a quick recap on all types of hyperlipoproteinemias. How many types of hyperlipoproteinemias? 6 types. Type 1 till type 5. But you have 2A and 2B. What are the 3 categories I told you? Elevation of only cholesterol. Second category, elevation of only triglyceride. Third category, elevation of both cholesterol and triglyceride. If cholesterol alone is elevated, they present with tendon xanthomas and accelerated atherosclerosis. If triglyceride alone is elevated, then you see eruptive xanthomas, recurrent pancreatitis. On examination, you find lipemia retinalis. If both are elevated, you see both features. Okay. Tell me which type fits into the first category. All of you answer. In elevation of cholesterol alone, it is type 2A. That is why type 2A is otherwise called as familial hypercholesterolemia. It is caused by the defect of LDL receptor defect. It is an autosomal dominant condition. Only triglyceride levels are high in type 1, type 4 and type 5. Type 1 is familial chylomicronemia syndrome. Type 4 and type 5 are familial hypertriglyceridemias. In type 4, only VLDL levels are high. In type 5, both chylomicron and VLDL levels are high. Okay. If both are elevated, then it includes 2B and 3. What did I tell you about 3? 3 is E defect. And in type 3, you see a broad beta bands. Hence, it is called as broad beta disease. So, what is the right answer here? The right answer is familial hypertriglyceridemia. A young boy is having, can you all please focus on what we are discussing now? Yeah, I gave you my points. I would say it as 6th codon. Okay, how many ever times you say whatever you have seen so far. If I don't see a conclusive evidence, I will not change. Okay, and I will not make mistake also. Yeah, young boy is having difficulty in breathing while running. This means there are neurological manifestations and is unable to rise from squatting position. Again, neurological. And you see cherry red spot. So neurological manifestations with cherry red spot is Tay-Sachs disease. Okay. So answer is Tay-Sachs disease. And I would like to give you one mnemonic to answer this question. Cherry red spots are absent. Okay. Cherry red spots are absent in KGF. Did you all see that movie? KGF? Yeah. Kolar Goldfield. Here KGF stands for Krabs disease, Gaucher's disease, Fabry's disease. Krabs, Gaucher's, Fabry's, you don't see cherry red spot. So what is the right answer here? The right answer here is Tay-Sachs disease. Okay. So I'm going to give you few clues. Yeah, few shortcuts to be able to answer about glycosphingolipidosis. Okay, so first I'm going to tell you about few enzyme defects which are always confusing and the names of those disorders. Okay, let's first clarify that and then we'll discuss about which lipid accumulates where. Okay, so beta glucosidase defect, alpha glucosidase defect, beta galactosidase defect, alpha galactosidase defect. These names are quite confusing. Okay, so beta galactosidase defect causes GM1 gangliosidosis. Yeah, it causes GM1 gangliosidosis. It can also cause MarQ-B disease. Do you remember? Today I told you galactose is present in keratin sulfate. Galactosyl sulfatase defect causes MarQ-A. Beta galactosidase defect causes MarQ-B. Okay, and it also causes Krabs disease. So all these three answers are right, okay, because galactose is present in beta form in 3 structures. One is GM1 ganglioside, the other one is mucopolysaccharide keratin sulfate, third is it is present in myelin. To metabolize these galactose in beta form, you need the same beta galactosidase, but they are coded by different genes. The name of the enzyme remains the same, beta galactosidase coded by 3 different genes. So depending upon which enzyme is defective, manifestations will differ. If beta galactosidase, which is supposed to metabolize GM1 ganglioside, is defective, that causes GM1 gangliosidosis. If beta galactosidase, that is supposed to metabolize keratin sulfate, is defective, that causes MarQ-B. The one which is supposed to metabolize myelin is defective, that causes Krabs disease. So all of you repeat this. Beta galactosidase defect causes 3 names. What are the 3 names? GM1 gangliosidosis, MarQ-B disease and Krabs disease. Alpha galactosidase defect causes Fabry's disease. Okay, alpha galactosidase defect causes Fabry's disease. Beta glucosidase defect causes the most popular Gaucher's disease. Alpha glucosidase, I already told you, alpha glucosidase or acid maltase defect. What did we learn? Defect causes Pompey's disease. Okay, so I hope you have memorized it. I hope all of you have memorized it. So tell me, beta galactosidase defect causes GM1 gangliosidosis, Krabs disease, MarQ-B disease. Alpha galactosidase defect causes Fabry's disease. Beta glucosidase defect causes Gaucher's disease. Alpha glucosidase defect causes Pompey's disease. I hope it's clear to everybody. Yeah. So now, which lipid accumulates in which condition? Okay. So in GM1 gangliosidosis, obviously GM1 ganglioside accumulates. In Tay-Sachs and Sandoff, GM2 ganglioside accumulates. And that is why Tay-Sachs and Sandoff are collectively called as GM2 gangliosidosis. So in Tay-Sachs and Sandoff, GM2 gangliosides accumulate. Additionally, in Sandoff, globosides also accumulate. So how do you differentiate Tay-Sachs from Sandoff? In both the conditions, GM2 gangliosides accumulate, but in Sandoff, additionally globosides also accumulate. In sialidosis, GM3 gangliosides accumulate. That is why sialidosis is otherwise called as GM3 gangliosidosis. In Fabry's, globosides accumulate. Yeah, in Fabry's disease, globosides accumulate. In Gaucher's disease, cerebrosides accumulate. Okay, in Gaucher's disease, cerebroside accumulate. Can you repeat this? GM1 ganglioside obviously accumulates in GM1 gangliosidosis. GM2 gangliosides accumulate in Tay-Sachs and Sandoff. In Sandoff, additionally, globosides accumulate. Sialidosis, GM3 ganglioside accumulates. In Fabry's, globosides accumulate. In Gaucher's disease, cerebrosides accumulate. So these can be one-liners that are asked. Okay. Now, few shortcuts to remember about these glycosphingolipidoses. The first shortcut is no cherry red spot in KGF. I told you just now, right? No cherry red spot in KGF. So cherry red spots are not seen in Krabbe's disease, Gaucher's disease and Fabry's disease. Second shortcut is no mental retardation in GF. Usually we say girlfriends are smart. We say that, right? Girlfriends are smart. So no mental retardation in GF. What does GF stand for? Gaucher's and Fabry's. Okay. No organomegaly. Organomegaly is never a feature of Tay-Sachs and Krabbe's. Okay, no organomegaly in Tay-Sachs and Krabbe's. Okay, so now I'm going to tell you few facts about Gaucher's disease. So what do you know about Gaucher's disease? Which enzyme defect causes Gaucher's disease? Beta glucosidase defect. When beta glucosidase is defective, glucosyl ceramide or glucosyl cerebroside accumulates. Where does it accumulate? It accumulates in the membranes of RBCs and platelets. So when this enzyme is defective, RBCs and platelets get to have abnormal membrane. These cells with abnormal membrane get engulfed by macrophages. That causes refractory anemia and thrombocytopenia. Okay, so the child always presents with refractory anemia and thrombocytopenia. After it is engulfed by macrophages, within macrophages, cell membranes of RBCs and platelets accumulate like fibrils. This fibril accumulation causes crumpled tissue paper appearance. Yeah, what does it cause? It causes crumpled tissue paper appearance. So the history will always be classical. They always say a small child presents with refractory anemia and thrombocytopenia. To find out the cause of this bicytopenia, bone marrow biopsy was done. Histopathological examination of bone marrow biopsy reveals crumpled tissue paper appearance, and they always give you a slide when you see a blue color cell with fibrils. Yeah, blue color cell with fibrils, that is crumpled tissue paper appearance. And whenever there is refractory anemia, you know there will be compensatory erythropoiesis. And when there is compensatory erythropoiesis, bone marrow expansion happens that causes bone erosion. This bone erosion is responsible for bony pain. So the child always complains of bony pain. To find out the cause of bony pain, if you take X-ray of the distal end of femur, you take X-ray of the knee. In the knee X-ray, the distal end of the femur looks like an Erlenmeyer flask. Have you all seen a conical flask? So the distal end of the femur looks like a conical flask. That is called as Erlenmeyer flask deformity. Okay. And this erythropoiesis does not only happen in bone marrow, it also happens in other reticuloendothelial tissues. And that causes hepatosplenomegaly. Okay. And enzyme replacement therapy is available for Gaucher's disease. So if you know these facts about Gaucher's disease, that is more than enough. Can you repeat this about Gaucher's disease? Which enzyme defect causes Gaucher's disease? It is beta-glucosidase or beta-glucosyl cerebrosidase defect. When that enzyme is defective, glucosyl ceramide or glucosyl cerebroside accumulates in the membranes of RBCs and platelets, causing refractory anemia and thrombocytopenia. And to find out the cause of bone marrow biopsy is done, histopathological examination reveals crumpled tissue paper appearance. Compensatory erythropoiesis causes bone marrow expansion, bony erosion, bony pain. X-ray of the distal end of the femur shows Erlenmeyer flask deformity. And there is also hepatosplenomegaly. Enzyme replacement therapy is available. That's all about Gaucher's disease. Okay. So answer this question. A child presents with anemia, thrombocytopenia. On examination, hepatosplenomegaly is observed. The child also complains of bony pain. What is it? The light microscopical examination reveals crumpled tissue paper appearance. So it is Gaucher's disease caused by the defect of beta-glucosidase. So can you tell me about other choices? Alpha-glucosidase defect. Please tell me the condition. Alpha-glucosidase or acid maltase defect causes Pompey's disease. Alpha-galactosidase defect causes Fabry's disease. Beta-galactosidase defect causes GM1 gangliosidosis, MARQ-B disease. And the third one is GM1, the third one is Crab's disease. A 25-year-old male presents with hypercholesterolemia. His elder brother also had died of heart attack a few years back. LDL receptor defect was observed. Please tell me the right answer, everybody. What is the most probable cause? LDL receptor defect is familial hypercholesterolemia. It is type 2A. Good. A patient presents with arthritis and arthroscopy reveals blackening of the joints. Right? Blackening of the joint is ochronosis. Ochronosis is a part of alkaptonuria. Right? Ochronosis is a part of alkaptonuria. Alkaptonuria is because of a defect of which enzyme, all of you? Homogentisate oxidase. Okay, so all of you remember blackening of joint is ochronosis, which is a part of alkaptonuria. Alkaptonuria is because of homogentisate oxidase defect. Can you tell me about other choices, everybody? Branch chain keto acid dehydrogenase defect causes maple syrup urine disease. Okay, tyrosinase defect causes albinism. You know that, right? Tyrosinase is an enzyme which converts tyrosine to melanin. All of you repeat this. Tyrosinase is an enzyme which converts tyrosine to melanin, and tyrosinase defect causes albinism. Okay. So, do you all know that alkaptonuria is a tyrosine metabolism disorder? And there are multiple metabolic disorders related to tyrosine. I am going to help you learn about all tyrosine metabolism disorders. First, I am going to tell you the normal tyrosine metabolism. Then I will tell you about all disorders. Everything will be complete in the next 5 minutes, provided you put your heart and mind into it. Okay. So what is tyrosine? Tyrosine is hydroxyphenylalanine. Do you all understand this? Tyrosine is hydroxyphenylalanine. Every amino acid metabolism is initiated by a transaminase. So, one second. So, what will be the first enzyme to begin the metabolism of tyrosine? Tyrosine transaminase. And I said tyrosine is hydroxyphenylalanine. Alanine on transamination forms pyruvate. So hydroxyphenylalanine on transamination forms hydroxyphenyl pyruvate. So what is the first product that you get? The first product that you get is para-hydroxyphenyl pyruvate. If you have para-hydroxyphenyl pyruvate, the next enzyme has to be para-hydroxyphenyl pyruvate dioxygenase. So para-hydroxyphenyl pyruvate dioxygenase converts para-hydroxyphenyl pyruvate into homogenesic acid. Now you know the next enzyme. Homogentisate oxidase converts homogenesic acid into mallyl acetoacetate. Cis-trans isomerase converts mallyl acetoacetate into fumaryl acetoacetate. If you have fumaryl acetoacetate, the last enzyme has to be fumaryl acetoacetate hydrolase that cleaves fumaryl acetoacetate into fumarate and acetoacetate. So this is about tyrosine metabolism. Can you all quickly tell me about normal tyrosine metabolism again? I will tell you about all tyrosine metabolism disorders, type 1, type 2, type 3, alkaptonuria. Okay. So the first enzyme is tyrosine transaminase acts on tyrosine, converts it into para-hydroxyphenyl pyruvate. Para-hydroxyphenyl pyruvate dioxygenase forms homogenesic acid. Homogentisate oxidase forms mallyl acetoacetate. Cis-trans isomerase forms fumaryl acetoacetate. The last enzyme is fumaryl acetoacetate hydrolase, which forms fumarate and acetoacetate. Okay. Now, type 1 tyrosinemia is paradoxically caused by the defect of the last enzyme. So, type 1 tyrosinemia is because of the defect of which enzyme? Fumaryl acetoacetate hydrolase. And this type 1 tyrosinemia is otherwise called as hepatorenal syndrome. It is called as hepatorenal syndrome because the precursor fumaryl acetoacetate that accumulates gets converted to succinyl acetone. And this succinyl acetone is toxic to liver and renal parenchymal cells. When liver parenchymal cells are damaged, it presents as jaundice, hepatomegaly, hypoglycemia, liver cirrhosis, hepatocellular carcinoma. Suppose the history says a 10-year-old boy presents with massive elevation of AFP. Imaging shows primary in the liver. Then you should suspect type 1 tyrosinemia. So why is type 1 tyrosinemia called as hepatorenal syndrome? Because here the child presents with jaundice, hepatomegaly, liver is damaged. Jaundice, hepatomegaly, hypoglycemia, liver cirrhosis, hepatocellular carcinoma. Because kidney is damaged, they present with Fanconi syndrome and chronic kidney disease. Okay. And type 1 tyrosinemia also mimics porphyria. Okay. Type 1 tyrosinemia also mimics porphyria. Many a times type 1 tyrosinemic children get misdiagnosed as suffering from porphyria. Okay. And this type 1 tyrosinemia is characterized by cabbage odor of urine. Okay, type 1 tyrosinemia is characterized by cabbage odor of urine. Okay. Now, type 2 tyrosinemia is caused by the defect of tyrosine transaminase. How to remember this? Type 2 tyrosinemia, defect of 2 T's. What are 2 T's? Tyrosine transaminase. Type 2 tyrosinemia is caused by the defect of 2 T's, tyrosine transaminase. We have two eyes. So type 2 tyrosinemia is otherwise called as oculocutaneous syndrome. Type 2 tyrosinemia is otherwise called as oculocutaneous syndrome because here the child presents with painful corneal erosions and palmar hyperkeratosis. Yeah, the child presents with painful corneal erosions and palmar hyperkeratosis. That's about type 2 tyrosinemia caused by the defect of tyrosine transaminase, oculocutaneous syndrome. Okay. Type 3 tyrosinemia is caused by the defect of para-hydroxyphenylpyruvate dioxygenase. Type 3 is because of the defect of para-hydroxyphenylpyruvate dioxygenase. This condition presents with intermittent seizures and ataxia. Okay, they present with intermittent seizures and ataxia. Interestingly, gain of function mutation, loss of function mutation of para-hydroxyphenylpyruvate dioxygenase causes type 3 tyrosinemia. Gain of function mutation of para-hydroxyphenylpyruvate dioxygenase causes Hawkinsinuria. Okay, gain of function causes Hawkinsinuria. Hawkinsinuria is characterized by swimming pool odor of urine. Okay, swimming pool odor of urine. For this, you can just remember a hawk, right? A hawk flying above the swimming pool. So, Hawkinsinuria is characterized by swimming pool odor of urine. Then, homogenticide oxidase is defective. That is called as alkaptonuria. Okay, and you will have to know about alkaptonuria because alkaptonuria is very common in India, and that is why FMGE papers often have a question on alkaptonuria. So first tell me, what is alkaptonuria due to? Homogenticide oxidase defect. So what accumulates? Homogentisic acid accumulates. When homogentisic acid accumulates, it undergoes oxidation to form benzoquinone acetate. This benzoquinone acetate undergoes polymerization to form melanin-like fibrils. When these melanin-like fibrils accumulate in the cartilage, it causes cartilage destruction, and hence it presents with eucranosis. So they present with multiple intervertebral disc bulges or prolapses, multiple joints pain. They complain of elbow pain, knee pain, ankle pain. That is eucranosis because of accumulation of melanin-like pigment in the cartilage. When melanin-like pigment accumulates in the skin, that causes pigmentation of pinna, tip of the nose. Thenar and hypothenar eminences are also pigmented. When melanin-like fibril accumulates in mucous membrane, that causes Osler's sign. So if you ask which is the first sign of alkaptonuria, can you see this? What is this called as? It's called as Osler's sign. Osler's sign is brown or black pigmentation of sclera along the line of attachment of medial and lateral rectus. This is the first sign of alkaptonuria because of accumulation of melanin-like pigment in the mucous membrane. When this melanin-like pigment accumulates in urine, you see that urine turns dark on standing, right? So the first symptom will be my urine turns dark on standing. Okay, alkapton meaning black, alkaptonuria is black urine disease. So can you all repeat all about alkaptonuria? Yeah, alkaptonuria is a tyrosine metabolism disorder. All of you, don't forget this. It's a tyrosine metabolism disorder. It is caused by the defect of homogenticide oxidase. And what does it present as? It first presents as urine turning dark on standing. On observation, you see pigmentation of pinna, tip of the nose, thenar and hypothenar eminences are pigmented. And the person complains of multiple joint pain, which is called as eucranosis. On examination, you see brownish or blackish pigment of sclera along the line of attachment of medial rectus and lateral rectus, which is called as Osler's sign. Okay, so that is all about alkaptonuria. That is all about tyrosine metabolism disorders. So please tell me, tyrosine transaminase defect causes what? Please answer. two T's. Defect causes type 2 tyrosinemia. Very good. Please tell me which enzyme defect causes type 1 tyrosinemia. Type 1 tyrosinemia is because of the defect of fumarate acetoacetate hydrolase. Alkaptonuria is because of the defect of homogentisate oxidase. Type 3 tyrosinemia is because of the defect of para-hydroxyphenylpyruvate dioxygenase. Gain of function mutation of para-hydroxyphenylpyruvate dioxygenase causes Hawkinsinuria. Okay. Melanin is synthesized from which of the following amino acids? Didn't I tell you? Tyrosine in the presence of tyrosinase becomes melanin. So melanin is synthesized from tyrosine. So in this table, I'm going to help you to memorize specialized products that you can get from all amino acids. Okay. From phenylalanine, the specialized product that you get is tyrosine. Okay. From tyrosine, what are the specialized products that you get? From tyrosine, just now you told me melanin. Not only melanin, neurotransmitters. What are the neurotransmitters? Dopamine, norepinephrine, epinephrine. Okay, dopamine, norepinephrine, epinephrine. And then thyroid hormone. T3, T4, all these are derivatives of tyrosine. So what are the specialized products that you get from tyrosine? Melanin, dopamine, norepinephrine, epinephrine and thyroid hormone. From tryptophan, please remember 60 milligram of tryptophan is equivalent to 1 milligram of niacin. Niacin, vitamin B3 is synthesized in your body from tryptophan. From tryptophan you get serotonin. From serotonin you get melatonin. So what are the specialized products from tryptophan? Niacin, serotonin, melatonin. Okay. From arginine you get nitric oxide. Okay, from arginine you get nitric oxide. From glycine you get many specialized products. Okay, the first product is glutathione. From glycine we get glutathione. Glutathione is gamma glutamine cystinyl glycine. Glutathione is gamma glutamine cystinyl glycine. So from glycine we get glutathione. Not only glutathione, from glycine you get creatine. Okay, the mnemonic that you can use to remember amino acids that are used for synthesizing creatine. Yeah, what is the mnemonic for you to remember? GAME. What does GAME stand for? Glycine, arginine, methionine. Okay, glycine, arginine, methionine are the three amino acids necessary for creatine formation. So what is the mnemonic I gave to remember the amino acids necessary for forming creatine? GAME, glycine, arginine and methionine. Glycine is necessary for heme synthesis. Okay, glycine plus succinyl-CoA form ALA. That is the first step of heme synthesis. Glycine is necessary for creatine formation. No. It is necessary for purine ring synthesis. Okay, glycine is necessary for purine ring formation. Glycine is the most abundant amino acid of collagen. Okay, so collagen you know is a triple helix. Don't you know that? Collagen is a triple helix. And every polypeptide chain in collagen has 1000 amino acids. These 1000 amino acids are made up of repetitive units of glycine XY. So the most abundant amino acid of collagen is glycine. So that's about the specialized products that you get from amino acids. So tell me, from phenylalanine you get tyrosine. From tyrosine you get melanin, dopamine, norepinephrine, epinephrine and thyroid hormone. From tryptophan you get niacin, serotonin, melatonin. From arginine you get nitric oxide. From glycine, glutathione, creatine, heme, purine ring and collagen. Okay, so that is about specialized products obtained from all these amino acids? So we know the answer here is tyrosine. Nitric oxide is formed from which amino acid? Please answer. Nitric oxide is formed from arginine. This also you know, which is a precursor of niacin? Tryptophan. Heme is derived from which of the following amino acids? Heme is formed from glycine. What is the first step I told you? Glycine reacts with succinyl-CoA in the presence of delta ALA synthase to form ALA. Ammonia is transported from brain to liver in the form of which of the following amino acids? From brain, it is transported as glutamine. Okay. So, ammonia is transported from all tissues to liver. Because only in liver, ammonia becomes urea. Okay. And from all tissues, ammonia is transported in the form of glutamate. From all tissues, ammonia is transported in the form of glutamate. There are two exceptions. One is neuron, the other one is muscle. From neuron, ammonia is transported as glutamine. From muscle, ammonia is transported as alanine. Okay, so tell me, ammonia is transported from all tissues to reach the liver because only in liver ammonia becomes urea. The most common non-toxic form of ammonia is glutamate. Exceptions is neuron and muscle. From neuron, it is transported as glutamine. From muscle, it is transported as alanine. So what is the right answer here? It is glutamine. So finally, we have reached genetics. I hope all of you are alert and awake. Yeah. I need your 100% focus and attention here because understanding genetics is not that easy. And in this discussion, I have started from the first till the last. I've included all techniques. Okay. So please watch this carefully. A couple presented to the OPD with their 4-year-old child who has self-mutilating behavior and poor growth. Identify the condition and the enzyme involved in it. The moment you see self-mutilation, what should you think of? Lesch-Nyhan syndrome. I think all of you keep asking this. How long will the session go? I think until I finish. Okay, probably till 11:30. Yeah, until we finish genetics. Okay, so Lesch-Nyhan syndrome, and Lesch-Nyhan syndrome is caused by the defect of hypoxanthine guanine phosphoribosyl transferase. Okay, Lesch-Nyhan syndrome is caused by the defect of hypoxanthine guanine phosphoribosyl transferase. So we know the answer is choice A, but I'm going to tell you what happens when hypoxanthine guanine phosphoribosyl transferase is defective. I hope you all can see this. Hypoxanthine guanine phosphoribosyl transferase is an enzyme involved in purine salvage pathway, wherein when nucleotides undergo breakdown, individual nucleotides become hypoxanthine and guanine, and these hypoxanthine and guanine again regenerate inosine monophosphate and guanosine monophosphate. So this is a way by which we recycle all nucleotides. Okay, so when this enzyme is defective, tell me what would accumulate. Hypoxanthine would accumulate, guanine would accumulate. When these nucleotides accumulate, they undergo degradation to form uric acid. That is why these children present with hyperuricemia. They present with hyperuricemia and gout. Okay. Additionally, they present with self-mutilation. So how do we remember all these facts about Lesch-Nyhan syndrome? I have a mnemonic for this, Lesch-Nyhan, right? So L stands for what? L stands for lips are bitten, nails are bitten. Okay, L stands for lips are bitten, nails are bitten. E stands for elevated uric acid. E stands for elevated uric acid. S stands for self-mutilation. Okay. C stands for they present with chorioathetosis. Okay, they present with chorioathetosis. H stands for hypoxanthine guanine phosphoribosal transferase defect. Okay, Nihan. Nihan sounds like a man's name. Nathan, Nihan, man's name. So you can use that to remember that it is X-linked recessively inherited. Okay, Nihan, man's name. You can use that to remember X-linked recessive inheritance. Are you clear? So what is the mnemonic that you're going to have for Lesch-Nyhan syndrome? L stands for lips are bitten, nails are bitten. E stands for elevated uric acid. S stands for self-mutilation. C stands for chorioathetosis. H stands for hypoxanthine guanine phosphoribosal transferase defect. And it is X-linked recessively inherited. Okay. So identify the pattern of inheritance depicted in the pedigree chart. Okay. Okay. Let me help you see this entirely. So what is the right answer here? Pedigree. The answer, let me tell you the answer first. It is autosomal dominant. Okay. Why am I calling it as autosomal dominant? Whenever you're looking at the pedigree chart, okay, always look for a generation wherein both the mother and the father are affected. Okay, can you see that here? Here both the mother and the father are affected. If it is an autosomal recessive disorder, then both the mother and father have the defective allele. Both the alleles are defective. In that case, all their children should be affected. Right? Irrespective of however the combination is done, all their children should be affected. Do you get it? So when both the mother and father are affected, if all their children are affected, then you should suspect autosomal recessive disorder. Whereas if it is autosomal dominant disorder, even this could have caused disease in the mother and the father. Right? In which case, if this A and A combine, the child is going to be normal. So even when the mother and the father are affected, if not all children are affected, then it is autosomal. If not all children are affected, then it is autosomal dominant disorder. Do you get it? So the answer for this question is choice D, but I'm going to give you few hints, right? Few hints or clues for you to be able to answer an MCQ based on pedigree chart. Whenever you look at a pedigree chart, step number 1 is find out wherever the mother is affected. Okay, I want you to see if there is a difference in the pattern of inheritance when the mother is affected versus when the father is affected. And if you see, whenever the mother is affected, the children get the disease. Whenever the father is affected, the children do not get the disease. Then it is maternal inheritance. If it is maternal inheritance, it's a mitochondrial disorder. Do you get it? So what will you do? You find out whether there is a difference in the pattern of inheritance when the mother is affected versus when the father is affected. If you see only when the mother is affected, children are affected. When the father is affected, the children are not affected. Then it is maternal inheritance, which is mitochondrial gene defect. Point number 1. Okay. Point number 2. As I told you, find out in any place when both the parents are affected. If both the parents are affected and if all children are also affected, then it is autosomal recessive. Okay, can you see that here? Whenever the mother is affected, you see that the children are getting affected. Whereas when the father is affected, children are all normal. So what will you call it as? You will call it as mitochondrial inheritance. Okay, second point. In a family with affected mother and father, if all the next generation kids are affected, then it is autosomal recessive. Do you see that? Both mother and father are affected and all their children are affected. So what is it? It is autosomal recessive. Third point, both the mother and father are affected, but some of the children are not affected. We call it a skip generation, right? In that case, what is it called as? Autosomal dominant. Can you see that here? Both the mother and father are affected. This is the exact image that was given in FMGE paper. So both the mother and father are affected. Still one child is normal. right? Which means it is autosomal dominant inheritance. That is the third clue, third hint. Fourth hint, okay? You're seeing that mostly men are affected, right? You're seeing that mostly men are affected. And whenever the mother is affected, the son always gets the disease. There is most of male preponderance. Males are mostly affected, but whenever the mother is affected, the son definitely gets affected. That is X-linked recessive. Okay, why do you think so? Why do you think so? When the mother is affected, it means the mother has got two X chromosomes, and both the X chromosomes there is a defect. Only then she has got the disease. Okay, and the son would have gotten one of the two X from the mother, which is defective, right? So the other Y from the father. So there is a defect here, and the son is affected. Okay, so whenever there is male preponderance, you check the mother, see where the mother is affected. And if you see whenever the mother is affected, all sons are affected, then that is called as X-linked recessive inheritance. Okay. Can you see that here? Here the mother is affected. Okay. And you see that both the sons are affected. Whenever the mother is affected, both the sons are affected. Again, mother is affected, all sons are affected. Okay. So that is called as X-linked recessive. Now, last one, you see more of female inheritance. Females have disease more. Okay. And whenever the father is affected, whenever the father is affected, yeah, whenever the father is affected, he is going to, let me show that here. Yeah, so so what was I telling you? Yeah, whenever you see that there is more of female preponderance, and whenever the father is affected, he definitely transmits it to his daughters. Okay, that is called as X-linked dominant. Okay, because the daughter would definitely get the X from the father, and the father is affected. The defective allele has come to the daughter, and it is X-linked dominant. Even if one allele is defective, there is going to be presentation. In that case, it is called as X-linked dominant. Okay, so do you see that here? Mostly females are affected. Okay, and whenever the father is affected, he is always sending it to the daughters. Whenever the father is affected, he is always sending it to the daughters. Then that is called as X-linked dominant. Okay, so that is about inheritance. That is about pedigree. So what are the 5 points that I asked you to remember? 5 hints. The first hint is, see if there is difference in inheritance when the mother is affected and the father is affected. Only when the mother is affected, all children are getting affected. When the father is affected, the children are not getting affected. That is maternal inheritance, mitochondrial DNA mutations. Okay. Second, whenever the mother and father both are affected, and if you see that all the children are getting affected, that is autosomal recessive. Even when both the parents are affected, if you see that some of the children are not affected, then that is called as autosomal dominant. Okay. And then the fourth one, more of male preponderance, and whenever the mother is affected, sons always get it. Then that is X-linked recessive. More of female preponderance, and whenever the father is affected, he always transmits it to the daughters. Then that is called as X-linked dominant. Okay. So that is about pedigree based analysis. All of the following are Purine bases except, which is not a Purine base? Uracil is not a Purine base because uracil is a pyrimidine base. Yeah, because uracil is a pyrimidine base. So let me tell you about 2 types of nitrogenous bases. One is purine, the other one is pyrimidine. The basic difference is purine with a smaller name has a bigger structure. It has 2 rings and 9 atoms. Pyrimidines with a longer name have a smaller structure. They have single ring and 6 atoms. That's a basic difference. Okay. Now, what are the pyrimidine bases we have? The mnemonic for pyrimidine bases is cut. Okay, the mnemonic for pyrimidine bases is cut. What does cut stand for? Cytosine, uracil and thymine. So all of you tell me, what are the three pyrimidine bases we have? Cytosine, uracil, and thymine. And what are the purine bases? We have two types of purine bases, major purine bases and minor purine bases. Major purine bases include adenine and guanine, which we already know. Okay, major purine bases include adenine and guanine. Minor purine bases include hypoxanthine, xanthine, and uric acid. Okay, minor purine bases include hypoxanthine, xanthine, and uric acid. This is just an introduction to genetics. Okay, so two types of bases, purines and pyrimidines. The mnemonic for pyrimidine bases is CUT, cytosine, uracil, thymine. Major purine bases include adenine and guanine. Minor purine bases include hypoxanthine, xanthine, and uric acid. So what is the right answer here? All of you please answer. That's what I'm doing, right? I'm taking class and I'm doing quiz. All the following are purine bases except, yeah, uracil, which is a pyrimidine base. Answer this, please. The histone that is not present in a nucleosome is. All of you start answering. The histone that is not present in a nucleosome is H1 histone. Okay. So now I'm going to tell you about organization. Okay, using this MCQ, this is a previous year question. So using this MCQ, I'm going to tell you about organization, just an outline of what is gene and what is chromosome and all of that. Because only based on this, you'll be able to understand techniques. If you don't know this, if you try to learn about techniques directly, that is never going to be possible. Okay, so stay with me for the next 5 minutes to understand about organization. What is the first level? The first level is a nitrogenous base, purine and pyrimidine. The next level, this nitrogenous base becomes a nucleoside, right? Then this nucleoside becomes a nucleotide. When many such nucleotides get linked, we call it as a polynucleotide chain. When two such polynucleotide chains interact with each other, we call that as double-stranded DNA. Right? This is called as double-stranded DNA. When a long double-stranded DNA gets condensed with the help of proteins, that is called as a chromosome. Right? So whenever you draw chromosome with centromere, a short arm and a long arm, I want you to visualize it as a structure which is nothing but a long double-stranded DNA which has got condensed with the help of proteins. So whenever you say with pride that you have 46 chromosomes, you mean to say that you have 46 double-stranded DNAs. Okay. So when a long double-stranded DNA gets condensed with the help of proteins, that is called as a chromosome. And every chromosome has multiple genes. All of you please watch this carefully. Every chromosome has multiple genes. These genes on transcription, okay, these genes on transcription give rise to mRNA. What is the first process called as transcription? Genes on transcription give rise to mRNAs. These mRNAs on translation, right? These mRNAs on translation give rise to proteins, right? This is called as central dogma. So can you all repeat this? Yeah, I come to Apollo Hospital, Greaves Road often because we supply blood collection tubes there. Trivus, I don't know how many of you know, we also supply Crivida Trivus recipe panel kit. So I often visit Apollo Greaves Road. Thanks, Aravind. Happy to know. Okay. So tell me, nitrogenous base, what are the levels? From nitrogenous base, it forms nucleoside. From nucleoside, it forms a nucleotide. When many such nucleotides are linked, we call it as a polynucleotide chain. When two such polynucleotide chains interact with each other, that's called as a double-stranded DNA. When a long double-stranded DNA gets condensed with the help of proteins, that's called as a chromosome. Every chromosome has multiple genes. One gene on transcription gives rise to one mRNA. That mRNA on translation gives rise to protein. Okay. Now, Why do you want to condense a long double-stranded DNA to form a chromosome? It is because one long double-stranded DNA measures a few meters long. And such a long double-stranded DNA cannot be packed inside a nucleus, which is few microns in diameter. Okay, so you have to condense such a long double-stranded DNA into a condensed chromosome. And that is made possible by histones. Right, that is made possible by histones. So what are histones? Histones are basic proteins. When I say they are basic, I mean to say they are positively charged. How about the charges of DNA? DNA is negatively charged. So there is an attraction, right? There is an electrostatic attraction between positively charged histones on one hand and negatively charged DNA on the other hand, so that a segment of double-stranded DNA gets wound around histone. Yeah, a segment of double-stranded DNA gets wound around histone. This string on bead appearance is called as a nucleosome. Okay, this is called as a nucleosome. So what is a string here? The string is a segment of double-stranded DNA. The bead is the histone that is present in the center. The string on bead appearance is called as nucleosome. And we have 5 types of histones. What are the 5 types? H1, H2A, H2B, H3 and H4. Okay, we have 5 types of histones: H1, H2A, H2B, H3 and H4. Out of these 5 types, all except H1, the remaining 4 form dimers. When 4 form dimers, we call it as an octamer. So this octamer sits in the center, that is rich in positive charges. Histone octamer sits in the center, that is rich in positive charges, that attracts a segment of double-stranded DNA that gets wound around, and this is called as nucleosome. So which histone is not present in a nucleosome? H1 histone is not present in a nucleosome. And several such nucleosomes exist. Just imagine that you have taken a long thread. On this long thread, you have placed beads at various places. You have allowed the thread to wind around those beads. Similar to that, what have you done here? You have taken a long double-stranded DNA. On this long double-stranded DNA, you have placed histone octamers at various places. You have allowed the double-stranded DNA to wind around histone octamers. Okay. So this fibril is called as 10 nanometer fibril. Okay, there are multiple nucleosomes. These nucleosomes are connected by a linker fragment, and H1 histone is present only in the linker fragment. This fibril is called as a 10 nanometer fibril because the width is just 10 nanometer. In the next level, I take a segment of this 10 nanometer fibril. I allow it to get folded on itself. Do you understand this? I take a segment of 10 nanometer fibril. I allow it to get folded on itself. Now the width increases. This is called as 30 nanometer fibril. This is where you get the second level of compactness. Where do you get the first level of compactness? When this double-stranded DNA binds around histone octamer, you get the first level. When 10 nanometer fibril gets folded on itself, you get the second level of compactness. Now, I take this 30 nanometer fibril and I form loops on chromosome scaffold. In your nucleus, you have 46 chromosome scaffolds, one for forming every chromosome. This scaffold is nothing but a tubular protein. For every chromosome, there is a central scaffold or a central tubular protein on which you have formed loops with 30 nanometer fibril. This 30 nanometer fibril has formed loops on all 360 degrees. It has spiraled around it, and on several such spiraling, you get a highly condensed chromosome which has a centromere, a short arm and a long arm. Okay, so whenever you draw a chromosome with a centromere, a short arm and a long arm, what should you visualize it as? You should visualize it as a tubular structure that is present in the center. On this tubular structure, you form loops with 30 nanometer fibril on all 360 degrees. It has spiraled around it. This is how you get a highly condensed chromosome. Are you clear about this? Yeah. So this is about organization. Okay. So tell me which histone is not present in a nucleosome. H1 histone is not present in a nucleosome. Which of the following is true about replication? Okay. Can you please answer this question? Is it a conservative process? I don't know how I'll be able to finish all of this. Beta and alpha DNA. It is not beta and alpha. B type of DNA and A type of DNA. B type of DNA is the most common physiological form of DNA, whereas DNA on dehydration forms A type. Are you clear about the first difference? B type is the most common physiological form of DNA. B type of DNA on dehydration forms A type. Both B type and A type are right-handed helices. In B type, for every full turn, you find 10 base pairing. In A type, for every full turn, you will find 11 base pairing. Okay, so these are the differences that you need to know between B type and A type. Okay, I hope that I've covered that. Okay, so what is the right answer here? It's a bidirectional process. Replication is a semi-conservative process and it is a bidirectional process. Okay, let me explain this to you. What is replication? All of you start answering. Replication is a process by which from one double-stranded DNA, we synthesize two double-stranded DNAs. Are you clear about this? What is replication? Because it's the basis of PCR. The next question is all about PCR. Okay. So what is replication? It's a process by which from one double-stranded DNA, we synthesize two double-stranded DNAs. In this case, the parent double-stranded DNA undergoes unwinding. Yeah, the parent double-stranded DNA undergoes unwinding. After unwinding, each of the two strands acts as template based on which you synthesize two new strands. So if you look at the two daughter DNAs, do you see that in both the daughter DNAs, only one of the two strands is new. The other one is old, which has been conserved. Because every time you're conserving a part of the DNA, you call replication as a semi-conservative process. Okay. Why do you call replication as a bidirectional process? Because during replication, there is unwinding. When there is unwinding, something called as replication bubble is formed. Right? What do you call it as? You call it as a replication bubble. Once replication bubble is formed, all enzymes start acting from the center. From the center, one set of enzymes synthesize in one direction, another set of enzymes synthesize in the other direction. And that is why replication is a bidirectional process. So tell me all about replication. Replication is a semi-conservative process and it is a bidirectional process. So the answer here is choice B. Okay. So I see that number of students are leaving the session. And only because you don't have the patience to learn genetics. Genetics sounds difficult, right? If you don't cross the initial part of genetics, you will never understand any of the techniques. Yeah, this is not like cursing. This is about a fact. Okay, so you will have to have that patience to understand the introduction to genetics. Okay, so how many of our students are staying back? Okay, whatever you're doing is right. Stay put. Watch this. We are going to learn about techniques anytime soon. Those who left the session have lost it. Okay. DNA polymerase requires all except. So tell me, what are the requirements of DNA polymerase? DNA polymerase always needs a primer and it needs a 3' 5' strand to act as a template. It can never use 5' 3' strand to act as a template. Okay, so it requires all except choice D. It can never use 5' 3' strand as a template. Okay, let's not worry about that, Vandana, now. Okay, now your aim is only to understand concepts. Once your concepts are clear, however closer to distractors are, you will exactly know the right answer. Only when you memorize the right answer, on the day of exam, it will be confusing. Okay, so it is the time for you to learn everything conceptually. Please move out of factual learning mode. Okay, the moment you start memorizing DNA polymerase is always 3 prime 5 prime as a template, you will never answer it right. Okay, can you all please focus on the session instead of asking me questions? I want your 100% focus on what I'm discussing, please. Yeah, so DNA polymerases, I'm going to tell you about requirements and properties of DNA polymerases. The first requirement of a DNA polymerase is all DNA polymerases need a template strand to be already present. Yeah, they always need a template strand to be already present. Only then they can synthesize a new strand which is complementary and anti-parallel. They synthesize a new strand which is complementary and anti-parallel. And all your DNA polymerases can synthesize a new strand only in 5 prime 3 prime direction. Okay, any DNA polymerase on this earth can synthesize a new strand only in 5 prime 3 prime direction. If they can synthesize a new strand only in 5 prime 3 prime direction, what should be the direction of the template strand? The template strand should always be in 3 prime 5 prime direction. That is why I told you the first requirement of any DNA polymerase is they always need a 3 prime 5 prime strand to act as a template. Only then they can synthesize a new strand in 5 prime 3 prime direction. Second requirement is all these DNA polymerases need a primer to be already present. Only then they can elongate a DNA strand. So the second requirement is they always need a primer. Third requirement is they always need all 4 deoxynucleotide triphosphates. They always need all 4 deoxynucleotide triphosphates. Because what do? They just read the template strand. If on the template strand there is A, they can recruit T. They can only recruit T. They cannot synthesize T. So you should provide them T which they can appropriately recruit and link. Okay. So they need all 4 deoxynucleotide triphosphates. They always need magnesium or manganese to act as a catalyst. They always need a buffer to maintain the pH. Okay, so these are the requirements of DNA polymerases. Are you clear? Can you all start answering? What are the requirements of DNA polymerases? They need a 3 prime 5 prime strand to act as a template. They need a primer. They need all 4 deoxynucleotide triphosphates. They need magnesium or manganese to act as a catalyst. And they need a buffer to maintain the pH. Now do you understand this question? What is the right answer here? They never use 5 prime 3 prime strand as a template. If the RNA sequence is CAAGGU, what is the sequence of the template strand? This is an FMGE Jan 2024 MCQ, recently asked. Okay. So for you to answer this question, I'm not going to expect you to randomly give me answers. I want you to understand it conceptually and give me the right answer. For you to understand this, you will have to know the difference between replication and transcription. Right? The difference between replication and transcription. What is the difference? In replication, you synthesize 2 double-stranded DNA from a single double-stranded DNA. So in replication, when the parent double-stranded DNA unwinds, both the strands act as templates based on which you synthesize 2 new strands, right? Whereas what is transcription? Transcription is a process by which you synthesize from a double-stranded DNA, single-stranded RNA, right? What is transcription? It's a process by which from a double-stranded DNA you synthesize a single-stranded RNA. So even in transcription, the gene undergoes unwinding. After unwinding, only one strand which is in 3 prime 5 prime direction. Only one strand which is in 3 prime 5 prime direction will act as a template based on which your RNA polymerase will synthesize a new RNA in 5 prime 3 prime direction. Okay. This 3 prime 5 prime strand which acts as a template will be called as a template strand. The other strand, which is in 5 prime 3 prime direction, does not even take part in transcription. Why do you think the other strand need not take part in transcription? Because my aim is only to synthesize a single stranded RNA that is already done. So the other strand, which is in 5 prime 3 prime direction, does not even take part in transcription. Though it does not take part in transcription, you call the strand as a coding strand. Why do you call something which does not take part in transcription as a coding strand? Because this coding strand, because this coding strand will have the same polarity and the same sequence as that of the new RNA that is synthesized, except for the fact that T in coding strand will be replaced by U in RNA. Right, except for the fact that T in coding strand is replaced by U in RNA. If you're asking me why, it is because so long till now, this template strand and coding strand were complementary and anti-parallel. You unbound template strand from coding strand, and then you use template strand as a template, and you synthesize a new strand again complementary and anti-parallel. So the new strand will be very similar. The new strand will have the same polarity and the same sequence as that of the coding strand, except for the fact that T in coding strand is replaced by U in RNA. Are you all clear? Yeah. So tell me what happens in transcription. In transcription, the gene or the double-stranded DNA undergoes unbinding. After unbinding, only one strand, which is in 3 prime 5 prime direction, acts as a template, based on which you synthesize a new RNA in 5 prime 3 prime direction. That 3 prime 5 prime strand will be called as a template strand. The other strand, which is in 5 prime 3 prime direction, does not take part in transcription at all, but you call it as a coding strand. Okay. So using this as a knowledge, can you tell me what will you do if they give you template strand sequence and they ask you to find out RNA sequence? Can you all answer this? What if they give you template strand sequence? First, write the template strand sequence in 3 prime 5 prime direction. What is step number 1? Check the polarity in which template strand sequence is given to you. Irrespective of the polarity in which the template strand is given to you, you write it in 3 prime 5 prime direction, because you know template strand is working only in 3 prime 5 prime direction. Okay, and then you write complementary sequences, mark it as 5 prime 3 prime. That way you will never go wrong. Do you get it? So what will you do if they give you template strand sequence? Check the polarity. You write it in 3 prime 5 prime direction, write complementary sequences, mark it as 5 prime 3 prime. Okay. What if they did not mention polarity? Whenever they don't mention polarity, it means it is in 5 prime 3 prime direction. In genetics, there is a rule. The rule is you have to write any polynucleotide chain sequence only in 5 prime 3 prime direction. Do you get it? It can be any sequence. It can be template strand sequence, it can be coding strand sequence, it can be rRNA sequence, mRNA, tRNA, anything. Okay? Every sequence has to be written only in 5 prime 3 prime direction. So if polarity is not mentioned, it is implied that it is in 5 prime 3 prime direction. What will you do? You first write it in 3 prime 5 prime direction and then write complementary sequences. Mark it as 5 prime 3 prime. Are you clear? If it is not mentioned, it is 5 prime 3 prime. This is for template strand. What if they give you coding strand? If they mention that the question is coding strand sequence, be relaxed, right? Maintain the polarity, maintain the sequence, just replace T by U. If it's coding strand sequence, maintain everything, just replace T by U. Okay, so with that in your mind, look at the question here. All of you please look at it. If the RNA sequence is CAAGGU, have they mentioned polarity? They have not. So what is implied? It means it is in 5 prime 3 prime direction. So 5 prime CAAGGU is the RNA sequence. So tell me what will be the template strand sequence? The template strand sequence will be 3 prime 5 prime. For C, what is complementary, everybody? For C, complementary is G. For A, complementary is T. For A, complementary is T, G, C, C, A. Okay, so template strand sequences 3 prime GTT CCA 5 prime. Okay, but you have to write it in which direction? Only 5 prime 3 prime direction. So can you write it in 5 prime 3 prime direction? ACCTTG. So what is the right answer? The right answer is choice C. Did you all understand this? I see that many of you have chosen it as B. No. Only when you write it in 3 prime 5 prime it is B. Right? But here it is 5 prime 3 prime. So what is the right answer? The right answer is C. Okay. So even before I finished this session here, I see that on Telegram group people are asking for notes. Okay, which means I have not watched this session. All of you, if there is a reason why we conduct this live, okay, when there is a live session, please watch it live and then later you can use the note as a backup. You definitely will not understand anything by looking at these notes. Okay, so watch the session and then use notes as a backup. A patient presents with multiple colonic polyps and has been diagnosed with colorectal cancer. I'm happy that you feel it is like a cakewalk. Yes, it will be a cakewalk. Okay, so we'll continue till 11:30. I'll finish genetics. Okay, and if time permits, probably I will tell you about ABG also quickly. A patient presents with multiple colonic polyps and has been diagnosed with colorectal cancer. There is a strong family history of HNPCC. Okay, which DNA repair mechanism is most likely defective? Yeah, HNPCC or Lynch syndrome. It's a defect of HNPCC. You see double C, right? It's a defect of double M. Mismatch repair defect. HNPCC defect is mismatch repair defect. Okay, so answer is choice B. So let me tell you about 4 repair mechanisms and the defects of these 4 repair mechanisms causing disorders. Okay, the most common repair mechanism is base excision repair. The first one is base excision repair. The second one is mismatch repair. Third one is nucleotide excision repair. And the last one is double-stranded DNA break repair. Right? What are the 4 repair mechanisms? Base excision repair, mismatch repair, nucleotide excision repair, and double-stranded DNA break repair. Base excision repair defect causes mute YH associated polyposis. Okay. Defect of base excision repair causes mute YH. H stands for human. Okay, human mutant Y protein. Okay, so mute YH associated polyposis, which also presents with multiple polyp followed by colon cancer. Okay, mismatch repair defect. Just now I told you MM defect causes CC, HNPCC. Okay, nucleotide excision repair. Excision has got X. Defect causes X, xeroderma pigmentosa. Nucleotide excision repair defect causes xeroderma pigmentosa and cocaine syndrome. Okay, X, xeroderma pigmentosa and cocaine syndrome. Double-stranded DNA break repair. Double-stranded DNA break happens in the presence of ionizing radiation. And whenever ionizing radiation like gamma rays cause double-stranded DNA break, repair can be done in 2 ways. One is non-homologous end joining. The other one is homologous DNA repair. Yeah, the repair can be done in 2 ways. One is non-homologous end joining. The other one is homologous DNA repair. Out of these 2, the most common type of repair mechanism is non-homologous end joining. And this is done by Q helicase. Okay, it is done by Q helicase, not the alphabet Q, KU helicase. And it is a suboptimal way of repairing it. Okay, it's a suboptimal way of repairing it because in non-homologous end joining, there is loss of nucleotide sequences. Yeah, there is always loss of nucleotide sequences. So it is a suboptimal way of repairing it. Whereas this is the best way of repairing double-stranded DNA break, but it does not happen that commonly. Okay, and a defect of double-stranded DNA break repair causes 3 disorders. Those 3 disorders can be remembered using a mnemonic ABF. So what does ABF stand for? Ataxia telangiectasia, Bloom syndrome and Fanconi's anemia. Okay, ataxia telangiectasia, Bloom syndrome and Fanconi's anemia. To make it more complex, if they ask you of these disorders which is caused by the defect of non-homologous end joining, that is only the first one. Ataxia telangiectasia is caused by the defect of non-homologous end joining. Non-homologous end joining defect also causes severe combined immunodeficiency. Right? It also causes severe combined immunodeficiency. Now, Bloom syndrome, Fanconi's anemia and human breast and ovarian cancer. Okay, Bloom syndrome, Fanconi's anemia and human breast and ovarian cancer. All these are caused by the defect of homologous DNA repair. Okay, so that is about DNA repair mechanisms and defects. So defect of base excision repair causes mute YH associated polyposis. Mismatch repair defect causes HNPCC. And then nucleotide excision repair defect causes xeroderma pigmentosa and cocaine syndrome. Non-homologous end joining defect causes ataxia telangiectasia and severe combined immunodeficiency. Whereas homologous DNA repair defect causes Bloom syndrome, Fanconi's anemia, human breast and ovarian cancer. Okay. Okay. So finally about techniques. Okay. Starting from here, I will tell you about PCR. I will tell you about RFLP. I will tell you about cytogenetics as well. Okay. Various types of PCR also. Okay. So the annealing temperature of PCR is, what is your answer? Bloom syndrome is homologous DNA repair. What I said is right. Homologous DNA repair. Okay. Yeah, the answer is variable. Only one. Bhavana alone has answered it as variable. One other person, A, variable. Yeah, answer is choice D, variable. Okay. So let's learn about steps of PCR. Before we start discussing about steps of PCR, let me define polymerase chain reaction for you. Polymerase chain reaction is an amplification process, right? Wherein from one copy of double-stranded DNA, we keep getting multiple copies. So what is PCR? It's an amplification process. And to add to it, it is an in vitro amplification. Because in PCR, amplification happens in a small test tube. It is not happening within a cell. It is happening outside a cell in a test tube. So it is in vitro amplification process. Okay. And it is in vitro amplification of a desired fragment of DNA. In PCR, I am not going to randomly amplify something. I amplify only a desired fragment of DNA. So how do I manage to amplify only a desired fragment? By constructing primers. I choose primers in such a way that these primers are complementary only to the flanking sequences of the desired fragment. Suppose, what do I mean by flanking sequences? Suppose this represents the entire double-stranded DNA. Okay, but I want to amplify only a fragment of this double-stranded DNA. How will I specify the PCR process that I want only this fragment? By choosing primers. I know my DNA polymerase always needs primers, right? So how am I going to choose primers here? I choose primers in such a way that these primers are complementary only to the flanking or the terminal sequences of the desired fragment. So that when my DNA polymerase elongates the strand, it only elongates here. So I get exactly what I want. I hope it's clear to everybody. right? So define PCR. What is polymerase chain reaction? It's an amplification process. It's an in vitro amplification of a desired fragment of DNA made possible by choosing specific primers which are complementary to flanking sequences of the desired fragment. Okay. Now, what are the requirements of PCR? You know, PCR is a replication process, right? Amplification is replication. And we know what are the requirements of replication. The same will be the requirements of PCR. So what are the requirements of PCR? You need a template strand that needs to be amplified. First requirement is template strand that needs to be amplified. Second is DNA polymerase which amplifies. And then requirements of DNA polymerases. Please keep the chat box active. Tell me, what are the requirements of DNA polymerases? They always need primers. They always need all 4-deoxynucleotide triphosphates. They need magnesium or manganese to act as a catalyst. They need a buffer to maintain the pH. Right? And apart from this, so first tell me what are the requirements of PCR. Just finish this and then I'll tell you the last one. So any PCR will need the template DNA that needs to be amplified. It needs a DNA polymerase which amplifies. It needs primers, all 4-deoxynucleotide triphosphates, magnesium or manganese as a catalyst, and a buffer. Okay. And all this process should happen in a machine. That machine is called as thermocycler. So which is the machine that is used for PCR? It is called as thermocycler. Okay. So these are the requirements of PCR. Now what are the steps of PCR? I want you to follow the steps of PCR, imagining that you're, say, working on sickle cell anemia diagnosis, right? If you're working on sickle cell anemia diagnosis, you know sickle cell anemia is caused by point mutation of 6th codon of beta globin gene, right? Sickle cell anemia is caused by 6th codon mutation of beta globin gene. So for this, you want only the beta globin gene. Okay. Suppose these two lines represent the entire chromosome. And the red lines here, do you see red lines in the center? These red lines represent beta globin gene fragment, which includes the 6th codon. So how will I get exactly what I want? By subjecting this DNA to PCR. I know PCR is a replication process, and you know for any replication, step number 1 is unbinding, right? Step number 1 is unbinding. So in the first step, I have unbound the double-stranded DNA to form 2 single strands. This unbinding is otherwise called as denaturation. So tell me, what is the first step of PCR? The first step of PCR is denaturation, wherein I increase the temperature to 94 or 95 degree so that all these hydrogen bonds are broken and the double-stranded DNA is unbound to form 2 single strands. This is the first step of PCR. After denaturation, in the next few steps, my DNA polymerase is going to elongate new strands. And I know for my DNA polymerase to be able to elongate new strands, they need primers. So in the next step, I am going to add 2 primers. These 2 primers are so chosen that they are complementary only to the flanking sequences of the desired fragment. So in the next step, do you see that I've added 2 primers which are complementary to the flanking sequences of both the strands. When I add these 2 primers go and anneal to the flanking sequences. So this step is called as annealing step. Okay, this annealing step should be carried out at an optimum temperature of 5 degrees less than the melting temperature of primers. Please repeat this. What should be the temperature of annealing step? It should be 5 degrees less than the melting temperature of primers. So if you're asking me why, it is because if it's more than the melting temperature, primers do not go and anneal, they undergo denaturation, right? If it is too less than the melting temperature of primers, my primers will go and anneal somewhere else. If the annealing temperature is too less than the melting temperature of primers, my primers will go and anneal somewhere else. I don't want some non-specific fragment to be amplified. I want a specific fragment to be amplified. So it has to be just 5 degrees less than the melting temperature of primers. Suppose melting temperature of this primer is 60 degree. Melting temperature of this primer is 62 degree. Then the average is 61 degree. 5 degrees less than 61 will be 56 degree. So for this combination and this example, annealing temperature will be 56 degree. But it varies based on the melting temperature of my primers. Melting temperature of my primers will vary based on the sequences, right? Suppose my one primer's melting temperature is 65, the other one is 67. In this case, the average melting temperature being 66, 5 degrees less than 66 will be 61. In this case, annealing temperature will be 61. That is why I told you annealing temperature is variable. It varies based on the melting temperature of primers that you have chosen. It is 5 degrees less than the melting temperature of primers. Do you get it? So after annealing, in the next step, my DNA polymerase is going to elongate new strand. And for this elongation step, I need a thermostable DNA polymerase which can withstand cyclical rise of temperature to 94 or 95 degree. Because whatever I am telling you now is just one cycle. In one cycle, there is denaturation where it is carried out at 94 or 95. Then there is annealing. Then there is elongation. Again, there is denaturation, annealing, elongation. So the DNA polymerase that I use for PCR should be thermostable. It should be capable of withstanding cyclical rise of temperature to 94 or 95 degree. That is why I have extracted DNA polymerase from an organism which lives in hot springs. And that organism is Thermus aquaticus. What is the name of the organism? Thermus aquaticus. If it can survive in a hot spring, it means it is thermostable. And the DNA polymerase that is extracted from Thermus aquaticus is Taq DNA polymerase. And the optimum temperature required for Taq DNA polymerase to elongate a new strand is 72 degree. And that is why elongation is carried out at 72 degree. Okay, so tell me what are the 3 steps of PCR? Can you all answer this? The 3 steps of every cycle of PCR would be number 1, denaturation, which is carried out at 94 or 95 degree. Annealing, which is variable, 5 degrees less than the melting temperature of primers. Elongation is always carried out at 72 degree. This is one cycle of PCR. This way, if you allow PCR to go through n number of cycles, at the end, from just one copy of double-stranded DNA, you can expect 2 to the power of n products. Such a sensitive technique it is. And the machine that is used for this purpose is called as thermocycler. Right? Now tell me, why is it called as thermocycler? The machine does not do anything. The machine does not do anything else. All that it does is it cycles between temperatures. First it increases to 94 or 95. Then it reduces to the annealing temperature that you have decided. Then it increases to 72, again increases to 95. It only cycles between temperatures. That is why the machine is called as thermocycler. I hope it's clear. So what is answer? The answer is variable. Can you answer this? We know the answer already. All the following are requirements of PCR except. Yeah. Restriction enzyme is not needed for PCR. Restriction enzyme is needed for RFLP. Okay. Answer is D. The technique used to quantify nucleic acid is. Please give your answer. Make a guess. The technique used to quantify nucleic acid is RT-PCR, qPCR, nested PCR, allele-specific PCR. The dark medico is right. Rahul Chaudhary, you're right. Good. Yeah, the answer is qPCR. qPCR or real-time PCR. Okay, real-time PCR or quantitative PCR. qPCR is quantitative PCR. It's also called as real-time PCR. It is used for quantifying nucleic acid. The right answer is choice B. Now, what is RT? The first RT PCR, reverse transcriptase PCR. Whenever you call something as RT-PCR, it is not real-time PCR. What is it? It is reverse transcriptase PCR. Okay. And when will you use reverse transcriptase PCR? When the nucleic acid of interest to you is an RNA. When the nucleic acid of interest to you is an RNA. For example, SARS-CoV-2 diagnosis. For diagnosing SARS-CoV-2 from the swab sample, all that you can get is RNA. Right? This RNA cannot be amplified. This RNA cannot be directly quantified. So what I should do is, before amplification or quantification, I should convert RNA to DNA. For that, I need reverse transcriptase PCR. All of you tell me, what is transcription? Transcription is DNA to RNA. So what is reverse transcription? RNA to DNA is reverse transcription. So using reverse transcriptase PCR, I convert the RNA to DNA and then I quantify it. For that quantification, I use real-time PCR. Do you get it? And then about the next choice, qPCR or quantitative PCR. So qPCR or quantitative PCR is a very new concept, right? We got it just 11 or 12 years back. Until then, we were using only conventional PCR even for quantification. So what we used to do is, we used to allow the PCR to get over. Yeah, this is PCR kinetics graph. What we would do is, we would allow the PCR to get over. After amplification is over, we would quantify the nucleic acid that is present here. From here, we would extrapolate to how much would have been there initially to give rise to so many copies at the end. After quantifying it here, we would extrapolate to how much would have been there initially to give rise to so many copies at the end. But later we found out that irrespective of the number of copies of DNA present in the initial sample, all these samples gave rise to same number of copies at the end. So from here, if you're trying to extrapolate, you will either underestimate or overestimate the number of copies of DNA present in the initial sample. So this is not the right phase for quantification. Which is the right phase for quantification? The initial lag phase or exponential phase. We call this phase as initial lag phase or exponential phase. This is the right phase for quantification. And real-time PCR, okay, real-time PCR or quantitative PCR is so called because it quantifies nucleic acid real-time. As and when amplification is happening, quantification is also happening simultaneously. Right? That is why it is called as real-time PCR, and it quantifies in which phase? It quantifies in the initial exponential phase or lag phase. And for this quantification, it either uses cyber green. For this quantification, it either uses cyber green or it uses TACMAN probe. Just remember these two names. In real-time PCR or quantitative PCR, we either use cyber green or we use TACMAN probe. Okay. So that is about qPCR or real-time PCR. So what is the answer for this question? The answer for this question is qPCR. But what is nested PCR? What is allele specific PCR? Because often students ask me to explain this. Nested PCR is used to increase the specificity. Okay. When do you have to increase the specificity? Whenever you have a gene, whenever you have a gene which is present in multiple copies with minimal variations. For example, glutathione S transferase. Glutathione S transferase, we have GST-M1, we have GST-M2, GST-M3, and we have multiple isoforms of GST. But suppose I want to show that GST-M1 polymorphism causes oral cancer. That's a proven theory. Okay, GST-M1 polymorphism causes oral cancer. So I want to amplify only GST-M1 gene. I don't want to amplify M2 or M3, but I know they have similar sequences. So however specific my primers are, there is a high probability that it goes and amplifies something else which I don't want as well. In that case, I use nested PCR, wherein what we do is, look at this. So this is the target sequence. By target sequence, I mean GST M1 sequences. And this is an unwanted sequence which I don't want to amplify. So step number 1, I construct 2 primers. These 2 primers go and anneal to my target sequence, but they also go and anneal to unwanted sequences. So at the end of the first PCR, I have 2 products. One is an unwanted product, the other one is a target sequence. I subject it to the next PCR reaction. In the next PCR reaction, I use another set of primer which is nested within the previous set of primer. Do you understand this? The next set of primer is nested within the previous set of primer. So this is very specific with the target sequence that I want. So it is going to amplify only what I want. Do you get it? And this is how we try to increase the specificity. So when do you try to increase specificity? When there are multiple copies of the gene with minimal variations, however specific your primers are, there is a high probability of unwanted sequences also getting amplified. To avoid that, you specify the target sequence twice with 2 sets of primers, one nested within the other. That is called as nested PCR. Okay. Now, the last one is allele specific PCR. Allele specific PCR is used for directly diagnosing the disease. Okay. Previously, I was telling you, suppose you are involved in molecular diagnosis of sickle cell anemia. I said you want a fragment of beta globin gene which includes the 6th codon. So all that you have done as a result of the PCR here is you have a fragment of beta globin gene which includes the 6th codon. Till now you don't know whether the 6th codon is normal or abnormal. So normal conventional PCR does not directly give you the diagnosis. And normal conventional PCR will only give you the fragment that you want. After you get the fragment, you should check whether the 6th codon is normal or abnormal. So there is another step that is involved. To simplify it, if I want to make a diagnosis just at the end of PCR, then I do allele specific PCR. Okay, wherein what do? I choose primer in such a way that these primers are complementary to the mutation site. I choose primers in such a way that these primers are complementary to the mutation sites. And these primers anneal only when it is normal. They don't anneal when it is mutated. So when I get the product, I know it is normal. When I don't get the product, it is mutated, right? That is called as allele specific PCR. Do you understand this? Suppose this is the normal sequence. And this is the mutated sequence. What I have done is I have chosen primer in such a way that my primer is complementary only to the normal sequence. So only as long as it is normal sequence, it goes and hybridizes and it elongates a fragment, I'll get a product. Once it is mutated, my primer cannot bind, I do not get a product. So if I get a product, it is normal. If I don't get a product, it is mutated. Right? So that is the advantage of allele specific PCR. It is used to directly detect mutations. But we don't prefer this. We don't prefer this very much because PCR failure is not only because of the presence of mutation, it could be because of something else. Right? So I don't know whether it is because of my PCR failure or is it because of mutation. So it is very, in one step, making a diagnosis is very challenging, right? It's very risky. I might give false positive results. And that is why we don't commonly use allele specific PCR, but this is what it is used for. Okay. So I hope you have understood all types of PCR, right? So reverse transcriptase PCR is used whenever the nucleic acid of interest use RNA to convert RNA to DNA. Quantitative PCR or real-time PCR is used for quantifying nucleic acid. For example, to assess viral load or to assess bacterial load or to assess fungal load. Nested PCR is used to increase the specificity. Allele specific PCR is used to directly detect mutations. Okay. No, it is not ending. If at all somebody should be tired, who should it be? Can somebody give me an answer for that? If at all somebody should be tired here, it should be me, right? Yeah, I'm not tired, which means you all should be very active. Okay. So, which of the following techniques is used for molecular diagnosis of sickle cell anemia? Yeah, immunohistochemistry? No, because immunohistochemistry is based on antigen antibody reactions. Right, it's based on antigen antibody reactions. It is used for protein based diagnosis. It is not used for molecular diagnosis. Okay, so immunohistochemistry cannot be used. What did I tell you about qPCR? Quantitative PCR only for quantification. Sickle cell anemia, there is no difference in quantity, there is difference in quality, right? So qPCR cannot detect. Reverse transcriptase PCR is used only when RNA is of interest, right? But sickle cell anemia is a mutation of DNA, right? So it is not RT-PCR. So what is the right answer here? The right answer here is RFLP. Okay, all of you, as I already requested, please watch what is being discussed. Don't always be in the fear of missing out mode. Yeah, FOMO does not help you. When I'm teaching this, you're asking about something else. Okay, please watch this, please focus on this. Till today, you did not understand all this. Today, I've understood, right? So, first, that happiness should prevail in your mind. Okay, that happiness helps you in becoming successful. Okay, it is not always about what I have not got. You should always be happy about what you have got. Okay, so the answer here is RFLP. Okay, so let me tell you about steps involved in molecular diagnosis of a known mutation site. Okay, so what do I mean by known mutation site? Example is sickle cell anemia. Sickle cell anemia all over the world is caused by point mutation of the 6th codon of the beta globin gene. You know that, right? Sickle cell anemia is caused by point mutation of the 6th codon of beta globin gene. Do we ever say in somewhere else it is caused by 11th codon mutation, somewhere it is caused by alpha globin gene mutation? No. So it is always fixed. That is when you call it as a known mutation site. So molecular diagnosis of known mutation site, always step number 1 is sample collection. Okay, and the sample that I always prefer is blood sample, because the DNA yield from the blood sample is really good. So I always prefer a blood sample. From the blood sample, next step is I extract DNA. Okay, then I say I've extracted DNA from a blood sample. I mean to say I've extracted all 46 chromosomes of the individual with all 25,000 plus genes. Right, that is what I mean. Suppose I get your blood sample. From your blood sample, if I extract DNA, it means in my hand I have all your 46 chromosomes with all your 25,000 genes. But do I have to work with so much of genomic material when I know I have to look only at the 6th codon of beta globin gene? I don't have to struggle with so much of genomic material. So how do I get exactly what I want? I get exactly what I want by subjecting the DNA that I have extracted to PCR. So in the next step, I am subjecting the DNA that I've extracted to PCR, wherein I choose specific primers in such a way that I get a fragment of beta globin gene which includes the 6th codon. So that is done. As a result of PCR, what do I have? I have a fragment of beta globin gene which includes the 6th codon. Till now, I don't know whether the 6th codon is normal or abnormal. So how will I find out whether it is normal or abnormal? To find out whether it is normal or abnormal, the next step has to be sequencing. Right? A straightforward answer would be sequencing. When I subject the PCR product that I have obtained to a sequencer, the sequencer exactly sequences the entire length. I see the 6th codon. I know what is normal and what is mutation, based on which I can give the report. So the logical next step can be sequencing. But because sequences are not widely available and sequencing is not a cost-effective option, right? We always do RFLP as the next step. So it is either sequencing or it is RFLP. Okay, so what is RFLP? RFLP is restriction fragment length polymorphism, and in RFLP we use a restriction enzyme. What are restriction enzymes? These are enzymes which cut at specific sites. Okay, so I choose restriction enzyme. There are hundreds or actually thousands and thousands of restriction enzymes available in the market. I cannot randomly go and choose a restriction enzyme. I choose a specific restriction enzyme in such a way that it cuts the mutation site only when it is normal. Do you understand this? How do I choose a restriction enzyme? I choose a restriction enzyme in such a way that it cuts the mutation site only when it is normal. So depending upon whether it is cut or uncut, I will know the diagnosis. Okay, I'll explain this further. I told you following PCR, I have a fragment of beta globin gene which includes a 6th codon. The length of this beta globin gene fragment is 1.4 kilobase pair. Yeah, the length of this fragment is 1.4 kilobase pair. I know that MST2, right? I know MST2 is an enzyme which cuts at the 6th codon when it is normal. So if the 6th codon is normal, MST2 cuts it to give rise to 2 fragments. One of length 0.2, the other one of length 1.2 kilobase per fragment if it is normal. If it is mutated, MST2 cannot cut it, in which case the length will remain as 1.4 kilobase per. So this information I know. How will I use this information for diagnosis? So what I do is after PCR, I take this PCR tube. This PCR tube has got the beta globin gene fragment of length 1.4 kilobase pair. Into this PCR tube, I add MST2. I incubate it. During the incubation period, MST2 tries to cut it. If it is normal, it is cut as 1.2 kilobase pair. If it's mutated, it is not cut. With a naked eye, I cannot find out whether it is cut or uncut. To find out whether it is cut or uncut, I run these fragments through electrophoresis. Okay, I run these fragments through electrophoresis. What I have drawn here for you is a gel. What gel is it? Polyacrylamide gel. This gel has 3 wells. Right, it has got 3 wells. I have 3 patients. Patient A, patient B, patient C. I add patient A's restriction digest into well 1, patient B's restriction digest into well 2, patient C's restriction digest into well 3. And then when I run it through electrophoresis, I know longer fragments cannot move. Shorter fragments can move faster. Right? Person A's restriction digest, I see that it is not moving at all. Yeah, person A's restriction digest does not move at all and it stops here. I know if it stops here, its length is 1.4 kilobase pair. So all of you tell me, what if the length is 1.4 kilobase pair? It means it is not cut. If it is not cut, it means it is mutated. So person A has mutation. Okay. Person B's restriction digest moves and stands here. I know if it moves till this, the length is 1.2 kilobase pair, which means it is normal. Person B is normal. Now look at person C. You're going to give me the diagnosis for person C, everybody. Person C's restriction digest shows 2 bands. One corresponding to 1.4, the other one corresponding to 1.2. So what does it mean? One allele is uncut, the other allele is cut. Do you understand this? One allele is uncut, which means it is mutated. The other allele is cut, which means it is normal. So the person is heterozygous for the mutation. So what is the diagnosis for person C? Person C genotype is AS. The person has got sickle cell trait. The genotype for B is normal, AA. The genotype for person A is SS, sickle cell disease. So A is disease, B is normal, C is trait. Do you all understand this? Yeah. So this is how we make a diagnosis of known mutation site. Okay. So can all of you tell me what are the steps involved in detecting known mutation sites? Molecular diagnosis of known mutation site starts with sample collection. From the blood sample that is collected, you extract DNA. When you extract DNA, you have all 46 chromosomes with all 25,000 genes. But we want only beta globin gene fragment. To get the fragment that I want, I subject the DNA that is extracted to PCR. Following PCR, I have a fragment, but I don't know whether it is normal or abnormal. To find out whether it's normal or abnormal, a straightforward answer would be sequencing. But sequencing is not a cost-effective option, so I subject it to RFLP. In RFLP, I choose a specific enzyme which cuts when it is normal. So if it is cut, it is normal. If it's not cut, it is mutated. I will know whether it is cut or uncut by running it through electrophoresis and by seeing the gel. Okay, so thanks, Ulagabi. I'm happy that you're all enjoying. So fish can be used to detect all of the following except. What is fish? Fish is fluorescent in situ hybridization that can be used to detect chromosomal aberrations. It cannot be used to detect point mutation. Yeah, it can be used to detect chromosomal aberrations. It cannot be used to detect point mutation. Okay. So whatever we learned so far is called as molecular diagnostics. Why do we call it as molecular diagnostics? Because we are diagnosing at the molecular level. What do I mean by molecular level? Not at the chromosomal level, not even at the gene level. Within the gene, nucleotide sequence alteration is what I am checking. One base alteration is what I am checking. So it is at the molecular level. But you know that some disorders are caused by chromosomal aberrations. So can you give me examples for chromosomal aberrations? Number one is copy number variation. What do you mean by copy number variation? Trisomy 21 or Down syndrome. Or it can be structural alteration like translocation. BCR-ABL translocation, right? So structural alteration. One is copy number variation, trisomy 21 or aneuploidy. Second one is structural alteration like translocation, example BCR-ABL translocation. Or it can be microdeletion or amplification of chromosome, right? Example is Prader-Willi syndrome. Yeah, Prader-Willi syndrome is caused by microdeletion of chromosome 15. Though it is called as microdeletion, a segment of chromosome is deleted. Okay, so what are the chromosomal aberrations you know of? Copy number variations or aneuploidy, translocation, microdeletion or amplification. To detect all these chromosomal aberrations, we use cytogenetic techniques. This is not molecular diagnostics, this is cytogenetics. So what are the cytogenetic techniques we have? Number one is karyotyping. Yeah, number one is karyotyping. Number two is fish. Number three is comparative genomic hybridization. Yeah, karyotyping, fish and comparative genomic hybridization. And out of these three, karyotyping is the simplest. Right? What is karyotyping? You must have seen karyotyping images for Down syndrome, right? Which is obtained by staining the chromosome with GM-SA. We call it as G banding of chromosome. You know that, right? What is G banding? It is GM-SA staining of chromosome. So in all these cytogenetic techniques, what we do is even here we collect a sample, and from sample we don't directly extract DNA, right? What we do is from the sample we extract WBCs, and this tissue culture is maintained, this cell culture is maintained. Why do we have to maintain cell culture for chromosomal aberration detection? Because I want metaphase arrest of the chromosome. Because only in metaphase the chromosomes are highly condensed. Only when they are highly condensed, staining will be easy and seeing the stained chromosome will be easy. Okay, so what do we do in cytogenetics? In cytogenetics, we collect the sample, WBC culture is maintained, metaphase arrest is induced, right? In that metaphase arrest chromosome, I paint all the chromosomes, and depending upon the number of bands that are seen, location of centromere, length of the short arm and length of the long arm, all these chromosomes can be arranged in a particular sequence. When I arrange these chromosomes in a particular sequence, if I see that there are 3 copies of chromosome 21, I know it is Down syndrome. So the take-home message for all of you here is karyotyping is done by metaphase arrest chromosome getting stained with GMSA, which is called as G banding, that can be used to detect only copy number variation or aneuploidy. Do you all understand this? It can be used only to detect copy number variation or aneuploidy. There is what is FISH. FISH is fluorescent in situ hybridization, wherein we use fluorescently labeled probe. Okay, and this fluorescently labeled probe can detect all types of chromosomal aberrations. How can it detect aneuploidy? For detecting aneuploidy, I use a probe which is a polynucleotide, and that is complementary to sequences of chromosome 21. And that I label with the fluorescent dye, right? That I label with the fluorescent dye. Then I take this probe, I take a sample from the patient, WBC culture is maintained, metaphase arrest is induced, and then I denature those chromosomes. Why do I denature those chromosomes? Because only then these probes can go and hybridize. So after denaturing those chromosomes, what I'm going to do is I'm going to add this probe. Then I add this probe, and when I look through a fluorescent microscope, in a normal person, I should be seeing only 2 signals, which means this person has got 2 copies of chromosome 21. But if I see 3 signals in the fluorescent microscope, it means a person has got 3 copies of chromosome 21, that is aneuploidy. Okay, so FISH uses fluorescently labeled probe, which are specific probes which are complementary to sequences of interest, and that can be used to detect aneuploidy. Okay, the same FISH can also be used to detect translocation. For example, if I'm treating a patient of leukemia and I want to find out if there is BCR-ABL translocation, because if there is BCR-ABL translocation, that's a good news. I have a targeted therapy for that translocation, right? So I want to find out if there is a BCR-ABL translocation, in which case I use 2 fluorescently labeled probes. One is complementary to BCR sequences. The other one is complementary to ABL sequences. And I differentially label them. BCR sequences are labeled with red fluorescent dye. ABL sequences are labeled with green fluorescent dye. Okay. And then I add it to metaphase arrest chromosome, which are denatured. Okay. And then I look through fluorescent microscope. What is normal is me seeing 2 red signals, me seeing 2 green signals coming from 4 different chromosomes is normal. This is normal, no translocation seen. Instead, if I see something like this, if I see something like this, it means BCR and ABL sequences are present in the same chromosome, right? BCR and ABL sequences are present in the same chromosome, which means there is translocation. Okay, so this is how we are going to detect translocation using FISH. Okay, and the same can also be used to detect microdeletion. Suppose I suspect Prader-Willi syndrome in a child. Okay, I will use a fluorescently labeled probe which are complementary to chromosome 15 fragment, which is expected to have undergone microdeletion in Prader-Willi syndrome, and I label it with a green fluorescent dye. I add it to metaphase arrest chromosome, which has undergone denaturation. And when I look through fluorescent microscope in a normal person, I should see 2 green signals. If I don't see 2 green signals, it means in one of the 2 chromosomes there is deletion. Do you understand? So one of the 2 chromosomes there is deletion, and this is how we find out microdeletion or amplification. Do you get it? So FISH can be used to detect all types of chromosomal aberrations. FISH can be used to detect aneuploidy, right? FISH can be used to detect translocation. fish can be used to detect microdeletion or amplification. So when can you not use fish? There are two circumstances where you cannot use fish. Fish cannot be used for molecular diagnostics. Then you are trying to identify one sequence alteration, because the probe's length 1,000 nucleotides. 1,000 nucleotide probe will not even know when one sequence is being altered. Okay, so it is a cytogenetic technique that can be used only for detecting chromosomal aberrations. It cannot be used for molecular diagnosis. It cannot be used for point mutations. Okay, another scenario where you cannot use fishes when you don't know what you're looking for. Right, when you don't even know what you're looking for. Only when you know you want to make a diagnosis of trisomy 21, you can send it to a lab asking for fish for diagnosis of Down syndrome. Right, only when you know you're suspecting Prader-Willi syndrome, you can send a sample saying Prader-Willi syndrome, because they are going to use specific probes. When you don't even know what you're looking for, there is a syndromic presentation, a child presenting with multiple facial dysmorphism, multiple cardiac defects, and multiple musculoskeletal defects, mental retardation. You don't even know what has caused this syndrome. In that case, don't ask for fish. The lab will send the sample back asking which probe to be used. Right? So when you don't know what you're looking for, fish cannot be used. In that case, comparative genomic hybridization has to be used. So for unknown chromosomal aberrations, okay, for unknown chromosomal aberrations, what do we use? We use comparative genomic hybridization. Okay. So we have learnt about cytogenetics also. So tell me the right answer. Fish can be used to detect all the following except point mutation. Do you want 5 minutes break? No. I don't know how long the session will continue. I think I'll stop with genetics today. Yeah. Probably in another YouTube session, I'll do ABG. Okay. Feeling sleepy now? Okay. We will finish the session after we discuss about RNA editing and after we discuss about lacoperon. With that, genetics is over. Okay. In liver ApoB gene, please watch FMGE session. Right, in that I have discussed vitamins, I have discussed minerals. Yeah. In liver, ApoB gene is completely translated to ApoB 100 and in intestine is translated to ApoB 48. Which of the following mechanisms explains this? The answer is RNA editing. Okay. So let me explain what is RNA editing. Before we start discussing RNA editing, understand that in RNA editing, you can edit only nucleotide sequences of an RNA. Okay, just remember that. Now about ApoB 100 and ApoB 48. Okay, so ApoB 100 is an apoprotein of VLDL. Okay, ApoB 100 is an apoprotein of VLDL. VLDL is formed in liver. Okay. Whereas ApoB 48 is an apoprotein of chylomicron, and chylomicron is formed in intestine. Are you clear about this? ApoB 100 is an apoprotein of VLDL. It is formed in the liver. ApoB 48 is an apoprotein of chylomicron, which is formed in the intestine. Okay, but interestingly, both these are products of one common gene, which is called as ApoB gene. And what we are trying to explain here is how can one gene give rise to different protein products in different tissues? How can ApoB gene give rise to ApoB 100 in liver and ApoB 48 in intestine? That is what we are trying to explain. So what happens is, in both liver and intestine, this ApoB gene undergoes the same transcription to give rise to the same mRNA. Okay, this mRNA leaves the nucleus and it reaches the cytoplasm. After it reaches the cytoplasm, there is a difference that happens only in intestine. Okay, enterocytes have an enzyme by name cytidine deaminase. So what is the enzyme that is present in enterocyte? It is called as cytidine deaminase. And what does a cytidine deaminase do? It acts on cytidine of a codon CAA, because the side is not visible. Let me write it here. Okay. Yeah. So both these are very similar. Even after they reach cytoplasm, they are very similar in liver and in intestine. After they reach the intestine, there is a difference that happens, because in both the cases, there is a codon CAA present in between. Okay, CA present in between. The enzyme that is present in enterocyte is called as cytidine deaminase, that deaminates cytidine of CAA to form UAA. When cytidine undergoes deamination, it becomes uridine. So CAA becomes UAA. And what is UAA? It's a stop codon. So only in enterocyte there is premature termination of protein synthesis. And enterocyte synthesizes a truncated protein. And this truncated protein has only 48% of amino acids that are present in the whole protein that is synthesized by hepatocyte. So this whole protein is called as ApoB100 and the truncated protein is called as ApoB48. Okay, this tells you how can one gene give rise to two different proteins in two different tissues. Do you understand this? So what did I tell you? ApoB100 is formed in liver, ApoB48 is formed in intestine, but both are products of one common gene which is called as ApoB gene. How can one gene give rise to different protein products in different tissues? What happens is both in intestine and in liver, the gene undergoes the same transcription to give rise to same functional mRNA. That functional mRNA reaches the cytoplasm. After they reach the cytoplasm, there is a difference that happens only in enterocyte. Enterocytes have an enzyme by name cytidine deaminase, which deaminates cytidine of a codon CAA to form UAA. So in enterocyte, stop codon is prematurely inserted. So in enterocyte, there is premature termination of protein synthesis and a truncated protein is produced. This truncated protein has only 48% of amino acids which are present in the whole protein that is synthesized by hepatocyte. So the whole protein 100% is called as ApoB100. The truncated protein 48% is called as ApoB48. Okay, so one gene can give rise to different protein products because RNAs undergo editing. When RNAs undergo editing, though the gene is the same, proteins can differ. That is why it is called as RNA editing. Do you understand this? So siRNA causes what? siRNA is small interference RNA. Okay, and it causes gene knockdown. siRNA causes gene, what is splicing? This is not splicing. Splicing is removal of intron and joining exon. Okay, I didn't talk about splicing. RNA editing is different from splicing. So siRNA causes gene knockdown. Let me tell you what it is. siRNA or small interference RNA interferes with gene expression. So what do you mean by gene expression? Gene expression is genes. Yeah, what is gene expression? Genes on transcription giving rise to mRNAs. mRNAs on translation giving rise to protein. This is called as gene expression. So the first process that happens is transcription. The second process that happens is translation. So whenever you say siRNA interferes with gene expression, you should mention whether it interferes at the transcription level or does it interfere at the translation level. The answer is it interferes at the translation level. How does it interfere at the translation level? These siRNAs have their own sequences. Small interference RNAs have their own sequences. Suppose they find complementary sequence on any of the fully formed mRNAs. Suppose these siRNAs find complementary sequences on any of the fully formed mRNAs. They go and hybridize with those mRNAs, forming a double-stranded RNA structure. Right? Not knowing that a double-stranded RNA has been formed, a ribosome will start translating this mRNA. When it translates this mRNA, when it comes across a double-stranded RNA structure, that's a signal that is given to ribosome that this mRNA has to be fragmented. So this mRNA will be fragmented and this mRNA will no longer give rise to a protein. Do you understand? So mRNA will no longer give rise to a protein. So it down-regulates gene expression. Do you understand this? What does it do? It down-regulates gene expression. Hence, it is called as gene knockdown. I hope it's clear to everybody. I think I answered this question already, Worthy Doc. You can expect 15 to 17 MCQs from biochemistry. There is no point in keep telling that biochemistry is your weakest, right? No subject is hard, right? It is about the perception that you have. Have your mind clear and watch this LRR session again, okay? You will be able to understand all aspects of biochemistry. From the beginning, from the first year, many of you think that biochemistry is dry. You think biochemistry is not interesting, or you think biochemistry is not essential. In all these cases, your cortex will not store the relevant information, right? Your cortex stores only the information that you think is important for you. Okay, the moment you think it's not interesting, it is my weaker subject, please don't do that. The moment you think of any subject that way, your cortex will not store an information. So please remove all that thoughts from your mind. Have an open mind, watch this LRR again and watch previous LRR and then try to check the rapid revision videos. That by itself is more than enough for you to get more than 15 MCQs right. Is that clear? Good, Mama. Very good. Yes, it's actually very interesting. Biochemistry is very interesting. Okay. So siRNA interferes with gene expression at which level? Translation level. It causes down regulation of gene expression. So it causes gene knockdown. This is the last MCQ for today. Yeah. LAC A and LAC operon codes for what? This is an FMGE Jan 2022 MCQ. LAC A and LAC operon codes for thiogalactosyl transacetylase. Thanks, Priya Reddy. Answer is thiogalactosyl transacetylase. So let me tell you all facts that you need to know about LAC operon. Okay. LAC operon is used for lactose utilization in E. coli. The interesting fact that you need to know about LAC operon is it is not present in human genome. It is present in E. coli genome. And it is for lactose utilization in E. coli. For E. coli to utilize lactose, E. coli needs 3 enzymes. The first enzyme is beta-galactosidase. Second enzyme is lactose permease. The third enzyme is thiogalactosyl transacetylase. Please repeat this. If at all E. coli has to utilize lactose, E. coli needs 3 enzymes: beta-galactosidase, lactose permease, and thiogalactosyl transacetylase. And if you look at the structure of LAC operon, in LAC operon, you will see a linear array of 3 genes. What are the 3 genes? LAC-Z, LAC-Y, and LAC-A. LAC-Z codes for beta-galactosidase. LAC-Y codes for lactose permease. LAC-A codes for thiogalactosyl transacetylase. Okay, and all these 3 genes share a common promoter and a common operator. Okay, so have a look at this. I'm going to define operon. What is operon? Operon is the linear array of genes of all the enzymes that are related to a particular metabolic pathway that share a common promoter and a common operator. Don't you see that here? What is operon? Operon is a linear array of genes of all the enzymes that are related to a particular metabolic pathway that share a common promoter and a common operator. This is the definition of lac operon. This is the structure of lac operon. Okay. Now about fuel utilization by E. coli. E. coli prefers to use glucose. As long as there is glucose, even if you add lactose, E. coli does not use lactose. Okay. Only when you deprive E. coli of glucose completely, only when you remove glucose completely from the medium, then E. coli goes through a lag phase and then it starts utilizing lactose. And the reason behind this way of fuel utilization by E. coli is because of the way lac operon works. For lac operon to be expressed, there are two requirements. The first requirement is cyclic AMP saturated cap. The first requirement is cyclic AMP saturated cap should go and bind to promoter locus. Okay, only then RNA polymerase can be recruited. So tell me, what is the first requirement for lac operon to be expressed? Cyclic AMP saturated cap should go and bind to promoter locus. Only then RNA polymerase can be recruited. Even after RNA polymerase is recruited, the operator locus has to be free. The second requirement is operator locus has to be free. But this operator locus is never free. Reason is, in lac operon upstream, there is a constitutively expressed LAC-I gene that is present. Right? Upstream to lac operon, there is a constitutively expressed LAC-I gene that is present. Constitutively expressed meaning it always expresses a protein. And the protein that is expressed by LAC-I gene is a repressor protein. This repressor protein tetramerizes and it always blocks the operator locus. It always blocks the operator locus. So, thanks. So tell me, what are the two requirements for lac operon to be expressed? One is you need cyclic AMP saturated cap to bind to promoter locus to recruit RNA polymerase. Even after that, for RNA polymerase to reach LAC-Z gene, operator locus has to be free. It is never free because LAC-I gene always produces a repressor protein that tetramerizes and it blocks the operator locus. Okay. Now, what happens in three scenarios that I told you? First, I'm going to tell you what happens when there is only glucose. Next, I'm going to tell you what happens when there is glucose and lactose. Third, I'm going to tell you what happens when there is no glucose and you add lactose. Okay. As long as there is glucose in the medium, E. coli happily utilizes glucose and the energy level of E. coli is high. As long as the energy level is high, adenylyl cyclase is suppressed. When adenylyl cyclase is suppressed, what is not produced? Cyclic AMP is not produced. So there is no cyclic AMP saturated cap to go and bind to the promoter locus. Furthermore, LAC-I gene product repressor tetramerizes that also blocks the operator locus. So both the requirements of lac operon to be expressed are not met. So lac operon is not expressed. And we also don't want lac operon to be expressed given the fact that there is no lactose in the medium. Do you understand this? When there is no lactose in the medium, why should E. coli spend its time and energy expressing lac operon? It need not be expressed. It is not expressed because both the requirements are not met. Okay. Second scenario, there is glucose and you are adding lactose. Okay. Now the same story. As long as there is glucose, energy level is high. Please continue. When the energy level is high, what is suppressed? Adenylyl cyclase is suppressed. So what is not produced? Cyclic AMP is not produced. So there is no cyclic AMP saturated cap to go and bind to promoter locus. But what can happen is, as long as there is lactose in the medium, this lactose by passive diffusion enters into E. coli and that lactose inhibits the tetramerization of the repressor protein. So what is free? The operator locus is free. Out of the two requirements, one is met, the other one is unmet. As long as both are not met, lac operon cannot be expressed. That is why I told you, along with glucose, even if you add lactose, E. coli continues to use only glucose, refuses to accept lactose because lac operon is not expressed. Okay. Now the third scenario. You're removing glucose completely and then you're adding lactose. Okay. When there is no glucose in the medium, what will happen to energy level? Decreases. When energy level decreases, adenylyl cyclase is stimulated. What is produced? Cyclic AMP is produced. Cyclic AMP saturated cap comes and binds to the promoter locus, recruiting RNA polymerase. Okay, and the lactose in the medium inhibits the repressor from tetramerizing. So operator locus is free. So RNA polymerase expresses LAC-Z genus beta galactosidase, LAC-Y genus lactose permease, LAC-A genus thiogalactosyl transacetylase. Then E. coli starts utilizing lactose. Are you clear? So these are the facts about lac operon. So what are the facts that can be asked about lac operon? They can ask you how many genes are present in lac operon. 3 genes. What are the 3 genes? LAC-Z, LAC-Y, LAC-A. LAC-Z codes for beta galactosidase, LAC-Y codes for lactose permease, LAC-A codes for thiogalactosyl transacetylase. Okay, and what are the 2 requirements for lac operon to be expressed? Cyclic AMP saturated cap should bind to promoter locus to recruit RNA polymerase, and operator locus has to be free. Both the requirements are met only when there is no glucose and there is lactose in the medium. Are you clear? So what is the right answer? LAC-A in lac operon codes for thiogalactosyl transacetylase. Okay. So with that, genetics is over. So we have discussed all about genetics starting from basic facts till applied aspects. In applied aspects, we learned about all techniques, various types of PCR. We learned about RFLP. We learned about cytogenetics. Yeah. So hope this helped. Yeah. Good night. Watch this session as many times as possible. In the sense, you can watch it again now and before examination also. For revising biochemistry, you can watch this. Yeah. My best wishes.